Good morning, everyone, or good afternoon. Welcome to our webinar series. This webinar will be on the use of MPS in drug development, and I will go through a little introduction really quick of the 3Rs Collaborative and this webinar series. First, I want to start with a disclaimer that I'm just going to read right off the slide. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither the 3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. All right, so I am Elisa Stevens. I'm the program manager here at the 3RC, and I wanted to welcome you all to this MPS on drug development webinar. Our Microphysiological Systems initiative seeks to increase adoption and regulatory use of MPS where scientifically justified, and we have over 49 member institutions and over 100 members, which you can see the logos here. We aim to provide thought leadership and expert consensus from developers combined with other stakeholders on the implementation of MPS, facilitate appropriate discussion, collaboration, and sharing between developers of MPS, create direct engagement between developers and regulatory agencies, develop external partnerships and collaborations to provide perspectives from developers throughout the field, and create resources to facilitate increased engagement with developers. We accomplish these goals through 3 work streams. One of those is the regulatory work stream, and we publish papers, host meetings, create presentations related to qualification of MPS systems. The Technology Expo work stream, which creates and maintains and promotes the Central Tech Hub, as well as a new publication hub. And then the End User and Education work stream, where we create educational resource hubs, webinars, and posts to advance MPS. With our End User work stream, in 2025, we've continued to do quarterly workshops. Those were AI and machine learning with MPS, retina and ocular MPS, multi-organ MPS, iSTAN program overview, and then now this one on drug development. We have posters at a variety of conferences and handed out flyers and pins for our members, and we continue to submit to all text corners. You can check out our previous webinars on our presentation page. If you go to MPS webinars under the events tab, you can find all of the previous webinars. Our regulatory group in 2025 did a publication on the perspectives on using MPS for regulatory applications, and you can look at that publication using this QR code. They promoted it at MPS World Summit and other conferences. They're continuing forward progress on the cross-platform DILI project, and excitingly, they're beginning to develop a benchmarking survey for MPS end users, and I think we will be disseminating that in January, so be prepared to respond. Our Tech Hub has 35 different MPS companies with various filters, and now we also have a publication hub that has over 300 MPS publications with filters as well. Today's workshop is focused on using MPS in drug development and regulatory submissions, and we wanted to note that this could include, but is not primarily focused on submissions to the FDA's iSTAN program. And we also wanted to remind you that MPS data can be submitted to the FDA without qualification through the iSTAN program. And hopefully we'll hear a little bit about that today. You can see our recent paper on 3Rs and regulatory applications with MPS, and our previous webinar on the iSTAN program is on our YouTube page and also on the events tab on our website. In today's webinar, we'll be using the Q&A function. So if you have any questions throughout the webinar, you can submit them by going to the Q&A tab on your Teams meeting. And we'll answer them at the end of the webinar. You can go to our website to learn more and join us to further the 3 Rs. And you can always email me if you're interested in joining our initiative or becoming one of our sponsors. All right, and now I will hand it over to David from IQ.
Thanks, Elisa. So my name is David Kukla. I'm a member of the IQ-MPS affiliate. So before getting started with this webinar, I just wanted to give kind of an overview of who we are. So the IQ is a collaboration of pharma and biotech companies, and it's created as an affiliate within the International Consortium for Innovation and Quality in Pharmaceutical Development. And the IQ-MPS has 4 main goals. One of them is to develop external partnerships and collaborations to help enhance the inclusion of industry priorities. Also to serve as a thought leader for MPS developers and stakeholder organizations. Also to provide a venue for appropriate cross-pharma collaboration and data sharing, and also to create focused engagement between industry and regulatory agencies on the current status and the evolving field of MPS. So the IQ is currently composed of 26 member companies with over 150 active participants, which represent experts from drug safety, 3 Rs, ADME, and PK/PD. So here I'm just showing our 2025 Executive Committee. Anya Kopek was our previous chair in 2024. Rhiannon David is our current chair. Kim Homan is the vice chair, and Philip Hewitt is the vice chair-elect. So here's just kind of a breakdown of how we structure the IQ-MPS. We have the Executive Committee, and they provide the leadership and guidance in the management of IQ-MPS business and affairs, and then helps implement strategic plans recommended by our steering committee. And then the steering committee is made up of 2 representatives from each member company, and basically they are the primary decision-making body for the IQ. Then within the IQ, we have 5 major work streams, and I'll kind of highlight those on the next few slides. But then within each one of these work streams, we have several different sub-teams. So the first work stream is regulatory engagement, and their mission is to build regulatory confidence in MPS and align on a common understanding of how MPS data should be handled in regulatory submissions by sharing industry knowledge of MPS utilization with regulators. The second work stream focuses on publications, and their mission is to outline the contemporary pharmaceutical industry perspective and considerations for developing, evaluating, and characterizing MPS models to support drug discovery and development. And this work stream has already published on several different organ systems and applications. I included several of those on the slides, but we are currently working on several more that will soon be published. Also, we have the pilot project workstream, which works to initiate near-term proof of concept studies for data sharing or prospective collaboration for qualifying MPS systems based on specific contexts of use. And then finally, the strategic partnership and communications workstream aims to develop external partnerships and collaborations to help enhance inclusion of industry priorities. Basically, by making the IQ-MPS the partner of choice for pharmaceutical MPS education, alignment, and engagement. And then additionally, the workstream disseminates IQ-MPS positions and learnings and influence external stakeholders. So if you have any questions, please feel free to contact us at the email provided on the slide, or you can keep up to date with IQ-MPS initiatives either on our website or our LinkedIn page. That's all I have. Thank you.
Thank you, David. All right, for our first presenter, I'd like to bring up Bruno from InSphero. If you could go ahead and share your slides.
Good morning. So, good morning or good afternoon, everyone, depending on where you're sitting. So I will kick off and present a case study for how liver microtissue are used in the drug development, including the regulatory process. Just going to take a pointer. So first part of my talk will be a quick presentation of the liver microtissue to give some context for those who don't know yet what that is. And the second one is a case study that was published this year, which used liver microtissue to de-risk dog finding and assess its relevance to humans. Yeah, so what are liver microtissues? So this is how they look. If you look at the little image in the center, it's a small spheroid. This spheroid is composed of primary human or animal hepatocyte or non-parenchymal liver cells. There is no extracellular matrix. You just see the cells as single cell suspension, and biology takes on and they organize themselves into this compact spheroid. We also use a chemically defined medium in order to control the culture condition precisely. As mentioned, you can make such small livers out of human donors, animal donors, from healthy population, or for vulnerable population, depending what your question is. Just growing hepatocyte and liver cells in 3D enable the cells to maintain their liver function much longer. This is not specific to liver microtissue. As soon as you grow hepatocyte in 3D, they don't tend to de-differentiate as it happens in 2D. They are also long-lived, so we still have liver function for these models after 28 days in culture. And the way they come is that in 96 or 384 well, one liver microtissue per well. So having this kind of physiological model is nice, but you need to be able to work with it in order to produce useful work. And for this, the scalability and the consistency of this platform comes along. So if you look at the ATP value of the volume of the microtissue across plate and across production, here each point is a different production, we can see that we have very stable ATP values and a very low coefficient of variation across plates, showing that the model are very similar across plate and between production. And having so many microtissues on a plate allow you to make very precise measurement, simply because you can multiply observation. So typically when we do a dose response curve, we can use 7 concentration, 4 replicate per concentration, and we get a very accurate measurement of the IC50, resulting in a very stable measurement across independent experimental run. So this is our positive control. We run it at every experimental run, and we get always the IC50 of chlorpromazine in the same range. So these models are being extensively used already by drug developer early in the pipeline. So we sold InSphero, just to give you a sense of how broad is the use, 300,000 of those human liver microtissue in 2024, 15,000 of the animal one. We work with over 55 drug developers, and we have a lot of publication describing or the use of this model. It is a selected list of the publication. The point is not to go through each of them, but if you get the slide afterwards, you can go through it and see which one might be relevant to you. So we are delving now into the case study. So a client, Sanofi to be precise, it was published at the beginning of this year, the story. We have a lot of such stories going on, but most of it never get published, which of course is a problem to promote the use of in vitro models for safety assessment. We have the chance that Sanofi was so kind to make a beautiful publication out of this. So this is the story I'm going to explain today. So it's about amsenestran, which was a selective estrogen receptor degrader, and it was tested in clinical trial in the context of breast cancer. However, the preclinical study was not so clean. So in the dog study, 1 month GLP and 3 month GLP, there was noticeable hepatotoxicity. The rat study was clean, even to the highest dose. And because this specific application tested was for cancer, nevertheless, you had the clinical trial starting because those patients would benefit enormously from such a drug. Nevertheless, it was very important for the regulator and Sanofi to clarify this dog finding. The first thing is to look at the exposure. So from the dose where we saw the hepatotoxicity, the lack of hepatotoxicity in rat was not due to a lower exposure. If we see the Cmax in the male and female rat, and also the area under the curve, they were largely superior to the dog, which showed hepatotoxicity. Here you have the values for comparison for the human in the plant tested dose, which are inferior to either species. So the first thing they did was to look at the metabolite. So this was not done in our model. It was done independently of that. And what they noticed first is that amsenestran is metabolized differently in the different species. And what they saw is that in the human and the rat cells, the first metabolite is M7, which is a phase 2 metabolite. In the dog, much less metabolism. However, from in vivo testing, they found that M5 was by far the prevalent metabolite. So they decided, they contacted us, and we started to do some testing on amsenestran and those two metabolites, M5 and M7, in human, rat, and dog liver microtissues. The first thing we do typically when we start from the get-go is a cytotoxic dose response curve. So it's really nice already to have a sense of the toxicity of the compound, but it's also a prerequisite for much more complex measurements afterwards for mechanistic study. The first observation is amsenestran. There is no obvious difference in cytotoxicity between the rat, the dog, and the human. And when you look at the M5 metabolite, which was the most abundant in dog, but also present in human and rat, we do seem to have more toxicity in the dog, but it's not really a very large difference. The M7 metabolite, which is the most abundant in human and rat, was not cytotoxic in any species. So the next step to try to understand the effect of amsenestran, specifically in dog, was to perform transcriptomic analysis, which is also something we do on a standard base at InSphero when working on drug safety. So since M7 was very clean, they proceeded only with amsenestran and M5. Following the transcriptomic study, the following was found for amsenestran. There was 2 different treatment time, 24-hour and 72 hours. And what we saw at the 24-hour time point is a very different reaction in dog and human for these selected gene sets that came as the most regulated. Principally, they are at odds. In dog, you do see a downregulation of phase 1 metabolism and also an inhibition of FXR, PXR, and a signature for hepatic cholestasis coming up. The opposite is true for human, where you have a strong upregulation of phase 1, upregulation of PXR, FXR, which are very important regulator of bile acid homeostasis, and the reduction of the hepatic cholestasis. The rat did not show any significant change on those gene sets. On the 72 time point, we still saw the downregulation of PXR and FXR in dog, and still the upregulation in the human. Looking at the M5 metabolite, we saw exactly the same thing for the dog with a very strong cholestasis signature and an inhibition of PXR, FXR, as well as phase 1, phase 2. The human was unaffected. The same is true at the 72-hour time point. with, again, hepatic cholestasis showing up in the dog and an inhibition of FXR, PXR, and the human showed just increase of phase 1 metabolism. So this is what we can see from the liver microtissue, but how is that relevant to the in vivo situation? So the next step was to test the hypothesis that those genes involved in the cholestatic response are indeed also regulated in the animals. And for this, they used frozen liver tissue from the preclinical studies to check for this. And they did qPCR in the in vivo male dog at 10 milligram per kilo per day, where no hepatotoxicity was measured. It was the dose higher where hepatotoxicity was noted. They selected the genes based on the one that were found to be regulated in liver microtissue, and they exactly saw a replication of what we saw in the liver microtissues. In blue, you have the control group. In red, you have the dog treated with 10 milligram. This is a log scale, so it's a profound inhibition of those genes. So principally, the in vivo testing corroborates the in vivo finding and gave confidence that what we saw in liver microtissue indeed is relevant for in vivo response. Therefore, the conclusion of the study, which satisfied the regulators, was that liver microtissues suggest that the liver injury in dog was not relevant to humans. And in good agreement with that, in the clinical trial for the oncology application, there was no hepatotoxicity noted in human later on. So typically what we have in the dog, we have a repression of FXR and PXR, which lead to a dysregulation of bile acid metabolism, resulting in a cholestatic liver injury. There is a kind of a mixed response in rat. However, for cholestatic liver injury, rat is typically not a good model because the bile acids are much more hydrophilic. In the human, we have an opposite response to the dog, where instead of being suppressed, you have actually an activation of FXR, PXR, resulting in a protective effect against the cholestatic potential of amsenestran. And this is the end of my presentation. I thank you for your attention, and I'll be happy to take on your questions.
All right. Thank you, Bruno. If we could have Leopold from TissUse share your screen, please.
Okay. Hi, everyone. I hope you can hear me okay. So my name is Leopold Koenig. I'm a senior scientist at TissUse, and I am going to talk today about how we modeled anti-IL-15 antibody responses in our human bone marrow MPS. First, as a disclaimer, so the opinions expressed today in this presentation are those of TissUse and not necessarily those of our partner in the study, Teva Pharmaceuticals. The compound that we were investigating in the study was a anti-IL-15 antibody. So IL-15 in general is involved in T cell and natural killer cell homeostasis, and therefore also is involved in immune-mediated diseases. Teva is developing an anti-interleukin-15 antibody to treat different inflammatory diseases such as celiac disease or vitiligo. And they developed a human antibody with a high affinity and potency for IL-15, where the efficacy was already demonstrated in animal models. So this compound is under ongoing clinical development. The idea, or what we wanted to do here in this in vitro bone marrow on a chip study, was to generate supportive data for a full toxicology package to support the initiation of clinical trials. And in the first in-human study, it was already shown that the antibody could lead to reduction of circulating NK cells in healthy volunteers. So the objective in the study was to determine the effect of the antibody on NK cell development in an in vitro human bone marrow model to set it up as kind of an in vitro safety study. And the tool that we used was the BiomimX bone marrow on a chip, which we further developed into a 3D human NK cell model to offer data that informs a clinical program. So we wanted to use this BiomimX bone marrow on a chip that we previously used or developed to basically study hematopoiesis in vitro. to mimic NK cell development in vitro as an advanced human relevant model for preclinical testing. Before we look into that specific case study, first some words about how this in vitro model of hematopoiesis works in general. So to show you the chip system that we used, you see on the left side in this image a representation of our chip. This is called the IQ-MPS chip 2. It is built by a glass slide on the bottom, a sandwich PDMS layer, and a polycarbonate adapter plate on the top. In that PDMS layer, there are two microfluidic circulations. So we have two technical replicates on one chip platform. And the system is completely closed. So it is the medium that is inside of the system is recirculated, enabling accumulation of growth factors and molecules inside of that media volume. Inside of each circuit, there are two compartments. One of those compartments we use in case of the bone marrow model for a zirconium oxide scaffold. You can see that here. So this is about the size of a 96 well plate. The zirconium oxide scaffold has an hydroxyapatite coating mimicking the spongious part of the bone. And we seed human mesenchymal stromal cells on there as a supportive cell type, and then the human bone marrow CD34 positive cells as the basis of human hematopoiesis in that system. The reason why we chose that setup is that with the addition of that ceramic scaffold, we can induce osteogenesis and niche expression or niche expression factors in the MSCs. So the MSCs basically cover the surface of the ceramic, build their own ECM, and then build an attachment surface and the right niche conditions for the HSC, so for the CD34 positive hematopoietic stem cells in that system. So this was also what these systems were initially developed for, to enable long-term cultivation of CD34 positive cells in vitro. And what we, before we initiated this study, already did in the past was to further develop that system into an in vitro hematopoiesis model. So we established a growth factor toolbox that would induce lineage differentiation in these systems. You can see a general representation of the assay up here. There's a 10-day pre-cultivation phase where we only pre-cultivate the stromal cells on the scaffold. And then with the seeding of the hematopoietic cells, the dynamic culture is started and then cultivated for multiple weeks. We would then take off at regular time points, so usually once or twice a week, cells for flow cytometry, and analyze basically the development of different hematopoietic lineages in the chip. With the addition of different growth factor cocktails, we can then basically target which lineages develop. So you see here, for example, with an erythroid cocktail, we see development of erythroid cells, not so much with a granulocyte cocktail or lymphoid cocktail. The other way around, for the granulocytes, those come up in a granulocyte mixture, not so much in the other ones, and the same for NK cells. But we can also combine basically these factors to get a more or less full picture of hematopoiesis in these systems. Normally, we can analyze all sorts of different lineages. So we usually analyze development of monocytes, neutrophils, reticulocytes, megakaryocytes, and NK cells. For this study, we focused specifically only on the NK cell part. So we didn't really look much on this whole lineage, but we really focused on development of NK cells in the system and put some work in characterizing this differentiation or this lineage a bit more in detail. In studies before, we were roughly looking basically on all different lineages and tracking NK cells only through one or two markers basically. Now we set up to understand a bit more in detail basically how different stages would develop. So we were first going into literature, looking basically which stages usually come up during NK cell development. For NK cells, these stages are classified into stage 1 to 5. And we were then selecting flow cytometry markers to being able to track these different stages of lineage development basically on chip. We saw quite early that along this NK cell lineages, also other cell types would develop without specific stimulation. So even when targeting NK cells, we would still have quite a lot of granulocytes and monocytes, as well as dendritic cells being present. But when looking into the NK cells, we were able to track basically all these different stages. So these stages are there at different proportions. There are some stages where the majority of cells reside in, like the stage 2B or stage 4, other stages where there are less cells in. But we could see that over the whole cultivations period, which was 35 days here, we were basically able to track all stages in the system. We could also see basically that we got quite far in development, so we were able to push these cells up into stage 5. The majority of cells, although stays in stage 4, though there seems to be some limitation how far these cells develop in vitro, but we could see that over time we also had a significant proportion of these late stage 5 cells, and those predominantly developed inside of the ceramic scaffold. So when we compare here the cells that we find inside of our ceramic scaffold model and the cells that circulate, we see that these later stages also developed first inside of the ceramic scaffold model. We also looked into whether these cells were functional or whether they could be activated. So we performed PMI ionomycin stimulation and saw upregulation of activation factor CD107A. And we could see that with or along the developmental timeline, the later stages, the more pronounced this activation behavior was in these cells. So more pronounced in stage 4 and stage 5 cells as expected. So now, after the general description of that model, let's switch over to the study of that anti-IL-15 antibody. So what we did first is we tried 2 different study arms with 2 different application schedules. So we tried a 1-week application, which you can see here from day 28 to day 35, and a 2-week application from day 21 to day 35, and compared it to an IgG control. What we saw is that we see a pronounced effect on circulating NK cell numbers. So as you can see here, basically we see a prominent drop of stage 5 and stage 4 cells here after 2 weeks, but also already after 1 week with a more pronounced effect after the 2 weeks. But we do not see pronounced effects on the progenitor stage cells, so stage 1 to 3 of the NK cell development. We were then also looking for these late stages, how those were affected, and we're checking the proliferation rate. What we learned is that there is a dose-dependent reduction of proliferation. So this further points in the direction that the antibody does not interfere with the early stages of NK cell development, but basically inhibits the proliferation rate of the stage 4 and later cells here, depending on the dose. We could reproduce this effect with different donors. You can see that there the donor react a bit differently. So there are different IC50 values between 2 different donors, but we always saw basically this pattern with different donors along the concentration range. We then also wanted to understand whether those effects were reversible. So after the 1-week treatment, which we then learned were already sufficient for a strong inhibition of NK cell numbers, we performed a 2-week treatment-free period. So basically, you can see here we have repeated application of the antibody from day 28 until day 35, and then a recovery phase where we removed the antibody and basically checked numbers over additional 2 weeks. We tracked here the stage 4 cells. What you can see here basically, so from day 28 to day 35, it's normalized on the day 28. You see the effect of the antibody basically here, depending on the dose. And then when looking into the recovery phase, we see that the effect of the antibody is quite potent. So you see that with the highest doses, the effect even is longer lasting. But then basically in the 2nd week, we see some beginning recovery with the lowest concentration already recovering after 2 weeks and going back to baseline values. Furthermore, we were looking into whether the cells were still functional under the treatment. So even if the cell counts were reduced, they were, of course, still remaining NK cells, and we checked whether we could still activate those. We learned there that the NK cells that remained were still functional, as shown here by the upregulation of the CD107 post in PMI ionomycin stimulation at all the time points tested, basically. So with that model, we could successfully characterize the effect of that anti-IL-15 antibody on NK cell development in a human bone marrow in vitro model. We could successfully trace NK cell development over several weeks and also demonstrate the functionality. We could show that the effect on NK cells is dose dependent, it's reversible and limited to the maturing NK cells without affecting the early NK cell progenitor stages. These findings are also highly consistent with the overall recognized role of IL-15 in NK cell development and are also in line with peripheral blood data from a clinical trial with the same antibody. So the results that we generated here were then used in an IND submission as a supportive data set, complementing the comprehensive toxicology package with in vitro human relevant data. With this, I would like to thank my colleagues here who worked on the project, our cooperation partners at Teva, and I guess we have 1 or 2 minutes also left for a question if there already is a pressing questions right now. Thanks.
Thank you, Leopold. All right, we have a question in the Q&A. From TissUse perspective, what kinds of validation frameworks or certifications will be more important to gaining regulatory acceptance of MPS and IND submissions? Which organizations are you engaging with or anticipate engaging with to shape these standards?
So I think for those, what is important is validation of these assays in ring trials or multi-center validations, and of course in close cooperation with the agencies such as FDA or EMA, which we are already also doing in some projects, basically, where we're trying to basically validate these assays better in ring trials with different pharmaceutical partners.
Great, thank you. All right, we'll go ahead and bring up Lorna from Emulate.
Oh, hello everyone. Thanks for the invite to be here. Happy to share some work that has been published and discussed previously. And the title of today's presentation is the use of an organ chip technology to support drug repurposing. So let me just move this off my screen because it's slightly distracting. Okay, it won't go. Okay, I'll have to leave it there. Drug efficacy and repurposing then. So slightly different perhaps to some of the prior presentations here, but nonetheless a very useful and important endeavour to accelerate new therapeutic or existing therapeutics that have been in the clinic. They're often designed to be safe. They were known to be safe, but sadly didn't meet the efficacy endpoints in their original indication. And being able to repurpose these, particularly in the case I'm going to describe, which was during the COVID-19 pandemic, it's a more rapid way of getting these drugs to the clinic. But clearly to go into the clinic, even though these drugs are often regarded as safe, they still have to demonstrate efficacy and therefore still have to go through a regulatory review. So this work was conducted using the Alveolix chip technology. So as a reminder for folks not familiar with the Emulate platform, you can see the chip here in the middle of the slide. We have a double channel chip where we have an upper and a lower channel. The upper channel is where in this situation we are placing primary human alveolar epithelial cells. They are a mixture of type 1 and type 2 cells. And actually in the paper that I will refer to, they do a nice job of showing that those cells both maintain their AT1 and AT2 phenotype. And this is actually a known phenomena with the correct microenvironment. So what you see is you see about a 50-50 split between type 1 and type 2 epithelial cells. However, you see that about 95% of the surface area is covered by type 1 cells, which again is exactly what we would expect to see in vivo. On the lower channel, we place primary human microvascular lung endothelial cells, and we situate these cells together for a couple of days. We then introduce flow, and we also introduce a mechanical stretch. So we do this particular model, I beg your pardon, just move back. We do this particular model on our chip S1, which is a PDMS-based chip, but that advantage allows us to apply this mechanical stretch that emulates breathing motions. This has also been discussed in prior literature as well that is published. The graph that you'll see on the right-hand side then indicates that not only in the presence of a liquid-liquid interface, which ultimately transitions to an air-liquid interface, and in the presence of this mechanical stretch, these cells can actually maintain a tight barrier between the epithelium and the endothelium. So the aim of the study that we're going to present today really looks at the infection of an alveolus epithelial chip with different viral infections. And this work actually started just prior to the pandemic in the lab of Professor Don Ingber. And the idea here was really to look at different flu strains. And the goal actually was indeed to try and identify either a drug that could be repurposed for flu, or indeed a new drug target that could be prosecuted in partnership with a pharmaceutical company. So it's been widely described and is known that these mechanical forces in the lung are very important, not just for its development. I mentioned about the maintenance of the type 1 and type 2 phenotypes. It's also important for surfactant production and maintenance of tissue barrier integrity. However, what's not known is the impact of these mechanical forces on the innate immunity, and thus how the lung itself responds to viral infections. So here, what we're looking at is a simple definition of what happens to the virus titer, as well as some cytokines in the presence of the breathing motions following stimulus with H3N2 virus. So you can see on the left-hand side that the virus titer goes down in the presence of breathing motions. This allows us to then stimulate these models in a more pathophysiologically relevant manner. You can also see that the cytokines trail and IP10 decrease. And if you look at the heat map on the right-hand side, what you'll see is that this is a time-dependent phenomena against the number of other cytokines and measures that you can see there listed. So in other words, to get maximum effect for this model, one needs to be running the model for about 14 days. What you'll also see is that in the static environment, which are the same cells cultured in the chip, but without any application of microfluidic flow or mechanical stretch, you actually don't see much change versus the day 0 chip outcome. So what the team did was, after day 14, they took the cells off the top channel and off the bottom channel. And here, what we're looking at on the left-hand side is a volcano plot of differentially expressed genes on day 14, following a 5% strain. These are genes, this is an RNA extraction from the top channel, the epithelium here. And if you wanted to understand a little bit more, there's some additional exciting targets that the team identified, but they were particularly compelled by this one here, S100A7, which sits in the top right-hand corner all by itself. So it's showing a very large fold change in the presence of the mechanical stretch after 14 days. So the team did a little bit of additional work looking at this S100A7 and found that the S100 family is a secreted protein family, often known as the alarmins, and it's known to bind to the RAGE receptor. And as a consequence of doing so, it can actually then alter some of the pro-inflammatory and pro-proliferative effects that are seen in the cell. And as such, this particular target has been explored in both neurodegenerative and cancer indications. The graph that you're seeing in the middle of the slide here shows the production of the protein. So this is actually measured in the effluent. So not only is there an upregulation of the gene, there's a functional consequence of that downstream, e.g. that the protein is actually secreted. So what the team did next was they went to the literature to find out if there was any clinically approved RAGE inhibitors. And as I mentioned in my prior slide, this is a target that has been prosecuted in a number of therapeutic areas, specifically for Alzheimer's disease. And if one looks in animal models, it's known that RAGE inhibitors, in particular this one, azelairagon, it had been taken into animals and was able to demonstrate a reduced A beta plaque formation within these particular animals. And it was therefore progressed into human clinical trials. It had a very safe profile. However, it did not meet clinical efficacy and hence was removed from clinical trial for Alzheimer's disease. So being able to access this particular drug, knowing it was safe, and by this time we were right into the middle of the COVID-19 pandemic. So it was deemed possible that this particular drug could be a new therapeutic for the cytokine storm that often occurs in the COVID-19 patient. So the team then took this drug and they looked at it in two different ways. So they looked at it in what was referred to in the paper as a prophylactic mode. So this is where they stimulate the chips with the virus, and then they go in and, I beg your pardon, another way around, they apply the drug to the chip, and then after about 48 hours, they're then infecting the chip with the H3N2 virus. And then they leave that for a period of 48 hours, and then they go in and they look at various different endpoints. They were looking at PCR for different gene changes, as well as using Luminex for some cytokine measurements as well. Now, many of you will be thinking, well, this is all fine, but this is not really what would happen in the clinical arena, and you're absolutely correct. So hold that thought, because we'll come to it in just a minute. But what you can see here in the graphs to the right-hand side are some key pro-inflammatory cytokines. And if you look at the control chip, the chip that's stimulated with virus, there's an increase in each of these pro-inflammatory cytokines. But in the presence of the drug here, azariligon, you can see that there's a reduction in each of these pro-inflammatory cytokines. And actually, it's these data here that were included in the clinical trial application to initiate the COVID-19 trials by a company called Cantex Pharmaceuticals. I should note that these trials are still active. They unfortunately have been slowed down quite simply due to fewer COVID-19 hospitalizations. Now, coming back to the importance of applying the drug at the right time and in a more disease-friendly context, the team did go back and look at the efficacy of this drug in what's known as therapeutic mode here. And here what they're doing is they're applying the drug at the same time as they are applying the virus. And to cut a long story short, as is the case with many antivirals, often you want to get them within about the first 48 hours of infection, not always possible. But moreover, it's been described many times over that the combination of different antivirals is actually where you drive efficacy. So in this particular experiment, the team actually used a different antiviral, molnupiravir, and this is also been known to be particularly active against coronaviruses, including SARS-CoV-2, as well as other RNA viruses such as influenza A. It's a broad spectrum drug, and when combined together with the azariligon, what you can see again is a very profound reduction in key cytokines, which would ultimately drive the clinical benefit. So in summary then, here using the human organ chips, these are microfluidic culture devices, allow us to recreate the tissue-tissue interface. In this particular example, we're recreating the alveolar capillary interface. We provide the infusion. We are in other circumstances able to permit immune cells into this environment as well. That was not done in this particular study, but we are able, with a combination of the microfluidic flow and the mechanics, to reconstitute that microenvironment. This allows us then to create an environment to more clinically model pharmacological activities, and we can perfuse particular drugs of interest at clinically relevant concentrations through the vascular channel. We found that the chip was able to maintain its vascular flow and air-liquid interface even during the mechanical stretching. And this model, in addition to the introduction of the flu virus, allowed us to rapidly test a new therapeutic and advance it into the clinic. So we would say that these systems do provide physiologically relevant test systems. And just before I end, there was a little bit of a mix-up around what I would be presenting today. Others have asked for an update on the Emulate's iSTAN progress, which, as many of you know, is a regulatory process. And this regulatory framework here, we had our letter of intent accepted in September of 2024. This was to evaluate our liver chip as a tool to de-risk compounds for drug-induced liver injury. On the right-hand side of the slide, you can read the press release. If you go onto the FDA website, you can see our letter of intent. You can also see our accepted qualification plan, which was accepted on the 8th of July of this year. And we're now currently in the final step of the qualification process, which is where we execute our plan, and we will submit next year our full qualification package. Once that's evaluated, if it passes, then we will be a regulatory qualified tool. And perhaps one final word on the iSTAN programme, just to draw your attention to this particular chart. It's a piece of work that a colleague in Australia is actually tracking, and it shows nicely all of the different letter of intents that have been accepted or rejected. So this first step is whether you have a reviewable letter of intent, and once it's deemed reviewable, you'll either get an accept or reject outcome. So you can see here the different programs, and I'll point you to the one at the top. It's a company called Integral Molecular. They have actually now submitted their full qualification package. So we fully anticipate that this will be the first company through the process, and we wish them well, and we hope to hear the outcome next year. So with that, I'll pause and I'll take any questions.
Thanks so much, Lorna. Did the regulators accept the alveolar model results as the main or only results to go into the clinic, or were there also in vivo studies?
No, they were the only data submitted into the application. There were no in vivo studies. As many people know, COVID-19 or SARS-CoV-2 doesn't translate well into preclinical species, particularly small animals. It will transition into primates, but here what we were looking at is the speed of entry into the clinic, and therefore I guess under the circumstances and the pandemic, that was one of the reasons why the in vitro data alone were sufficient. Please also remember it is a repurposed drug, and therefore a lot is known about this drug in terms of its safety and tolerability. So undoubtedly that will have helped the passage as well.
All right, great. Thank you so much, Lorna.
You're welcome.
Next we'll have Paola from BiomimX. So go ahead and share your screen.
So thank you very much for this opportunity, and hello everybody, good morning or good afternoon, depending on where you are. I'm Paola Occhetta, I'm CEO of BiomimX, and today I will describe you a study that we did in collaboration with the Swedish Biotech, Sinartro, that was part of the IND application for a new therapeutic that they are developing for the treatment of osteoarthritis. So before digging into the case study more deeply, just a couple of words on our technology. So in BiomimX, we propose beating organs on chip, which are models in which we combine two main aspects of many tissues in our body that are mechanical forces on one side and the recapitulation of three-dimensional architecture. And this is possible thanks to our patented technology, which is UBIT, that you see the mechanism of action in this video. Basically, we are able to apply a tunable mechanical stimulation that ranges from three-dimensional compression to three-dimensional stretching to a combination of both to cells embedded in a three-dimensional microenvironment. And our technology is versatile, so depending on the combination of the micro channels and of our mechanical stimulation, we can achieve different possible configuration, even working for three-dimensional barrier-like models. Why do we apply this combination of mechanical stimulation and mechanical environment on one side to make our tissue more mature with a higher level of differentiation, which is usually driven by mechanical triggers, and on our side, and this will be the focus of my case study today, also to drive pathological phenotype inside our model, which are specifically linked again to mechanical dysregulation. And this is indeed, and of course, applied to many different contexts of use, both related to safety, efficacy, and also personalized medicine. Thanks to our UBIT powered technology, we developed in the past many different biological models that were extensively scientifically and pharmacologically validated. And you can see the main one that we are working on in this slide. But now I want to really focus my attention on one of our main and mostly characterized model, which is a model of osteoarthritis-on-chip. And the main reason is indeed because I want to present the case study we built now, I think 2.5 years ago with Synarthro, which is a Swedish company that approached us because they were developing, and they are still developing, a new potential injectable treatment for osteoarthritis, which is the pathology of the cartilage joint, which I will introduce in a second. A couple of words on what Synarthro is doing and why they approached us. Basically, Synarthro came to us because they were working since a lot of years on an injectable therapy called the SYN-321, which basically combined a solid component, which is hyaluronic acid, which was covalently bonded thanks to a specific linker that they patented to a anti-inflammatory drug, which is diclofenac. And they showed nicely that by the use of this unique linker, they were able to make this link really strong and to promote a slow release of diclofenac into potentially into the joint. And this is the reason why SYN-321 has the ambition to provide a sustained pain relief and potentially an anti-inflammatory effect in order to promote its efficacy as a relief in pain for osteoarthritis, together with the well-known effect of hyaluronic acid, which is mainly the increase of joint lubrication. So when it came to us again, I think now mostly 3 years ago, they already performed a few studies of this SYN-321 that they were using to put together an application for phase 1 to a clinical studies. And in particular, they did some preclinical study in horses, mainly to show and to prove the slow release of diclofenac thanks to this specific linker that they patented. And they also performed several toxicological studies still in animal model that they give positive safety results for the new compound. And also they did some basic in vitro study to validate the hypothesis mainly on the slow release of diclofenac. So still not any efficacy test on actually the potential effect of this SYN-321 in treating osteoarthritis. And actually, this is why they came to us, because they had at that time a gap of knowledge. They wanted to better understand the mode of action of this therapeutic compound at the cellular and tissue level, potentially in a human and mechanically active relevant model of cartilage. And this is indeed to present you the reason why they approached us. Now, let me just do a brief description of our model at that stage. So why they came to us, because they were reading about our uni model, which is an osteoarthritis-on-chip setup. A couple of words on the pathology. So osteoarthritis is a complex disorder, is a disorder of the entire joint that causes disruption of many different components in the joint. And still a lot is unknown about the etiology of this disease, but what is known is that there is a strong correlation with mechanical dysregulation. And this is why we took the hypothesis that by recapitulating this mechanical dysregulation, we could actually reproduce osteoarthritis in an in vitro setup. So a few years before talking with Synarthro, we were already working on osteoarthritis. And here just a brief recap of the model we published in 2019. Starting from the hypothesis of mechanical dysregulation, we started to generate a model in which we culture chondrocytes in 3D, so in an hydrogen inside our model in static condition, and then we applied a hyperphysiological compression to induce the disease. Briefly, we demonstrated that we could actually achieve a nice and healthy cartilage inside our setup, inside our chip, that had traits that were resembling native cartilage that we use as benchmarking. And then we showed that by altering mechanically this environment, by applying a hyperphysiological compression in the model, we could actually induce many different traits of the osteoarthritis disease. And in particular, we could see how a mechanical dysregulation enhanced inflammation inflammation and degrading mediators such as MMPs and interleukin, inflammatory interleukin cytokines. And on the other side, it also promotes a loss of cartilage matrix component, which is indeed a hallmark of osteoarthritis in vivo. Last but not least, our mechanical dysregulation also changed the phenotype of our cells in the model by promoting the acquisition of hypertrophic traits, which is another characteristic trait of the pathology in patients. Now we also preliminary tested this model as a potential drug screening tool by using soluble molecules, and in particular we chose a couple of molecules that are known to have some anti-inflammatory and anti-degrading effect, and we showed how in our model subject to hyperphysiological stimulation we were able to see actually the effect of this compound in correlation with available clinical data, while the same was not possible in a model that was just induced by using more a standard approach like an inflammatory stimulation. And this was actually the state of our model when Sinatra approached us. And just repeat this slide just to remind you that they were not interested in testing small molecules, but in testing actually an injectable therapies. So this is the reason why when they approached us, the first thing we had to do together was to think about a redesigning of the model to be able indeed to allocate an injectable therapies inside. And their requirement were indeed to have their therapeutic product in direct contact with our cartilage microtissue and subject to the same kind of mechanical stimulation, because this is what it would happen actually in the joint. So this is the reason why we slightly modified our setup. Indeed, as I explained in the beginning, our platform is really versatile, so we were able to accommodate more than one channel and to have the space in between, the one that you see here in blue, to allocate an injectable therapeutic. And we proved, like by first technological validation, that that was feasible. Then we went on, and of course we just had to first re-qualify the model and prove that actually also with this technological modification still we could induce an OA-like phenotype inside the model, and basically reperforming the same assay that I showed you before, we demonstrated again that we were able to induce inflammation, degradation, and a phenotype switch inside of our new technological platform. And at this point, we were ready to start testing the SYN-321 therapeutic product. So first, we repeated the test that Sinatra did already in animal model to understand if we could indeed capture the slow release of diclofenac, as it was the case for animal studies. And actually, in our experiment, we discovered and we showed how a mechanically active environment enhanced the release of diclofenac, which may suggest that the presence of a mechanical stimulation may lead to a different or a more pronounced release of this drug. And then we came to the main test that was indeed to test and to understand which was the effect of SYN-321 at the cellular and tissue level inside a model in which we induced preliminarily osteoarthritis traits. And for this, we injected either SYN-321 or as a control only diclofenac and only hyaluronic acid to understand the effect of this unique combination. And actually what we studied first was the effect of this therapy on the inflammation and degradation of the matrix. And what we nicely saw was that SYN-321 exhibit an anti-inflammatory effect, which was bringing back the state of anti-inflammatory genes at the similar level of an healthy condition. And actually I want to stress that this was not the case by using only diclofenac or only hyaluronic acid. And also SYN-321 played a role in reducing matrix degradation. Similarly, effect that was not noted in the single component. This effect was also confirmed at the protein level. Here you can see an MMP13 staining showing how the MMP13 activity was switched off only in the case of combination of SYN-321, or mainly in the case of combination. Then we went on and we also checked the possible activity of SYN-321 in anabolic component, so to test if it was actually also able to restore an healthy cartilage. And actually this is still something that we are trying to understand. At the gene level, it seems not the case, so still either SYN-321 is not restoring any production of a normal cartilage, even if at the protein level we could observe an enhanced production of aggrecan in case of testing, of treating the system with the SYN321, but even with the control. So this is something still that is under investigation. So to conclude and give you some take-home message and conclusion, we successfully showed that we could develop actually a new technology to respond to the specific question of our sponsor. And of course, the new technology was qualified to prove it was compatible with the previous results. And thanks to this new technology, we could help SYNARTRO proving that SYN321 can decrease the inflammatory response typical of OA. It can slow down the onset of matrix degradation, and it can partially restore aggrecan production. And it responds to mechanical stimulation in terms of release of diclofenac, and it has no negative impact on cartilage markers that were still detected. And actually, this test helped them de-risking the transition to first in human by strengthening the mechanistic rationale behind the expected effect, a local effect of SYN321. And this data provided a human relevant mechanism of SYN321 efficacy. And I want to underline, of course, they were not the only data they were presented to regulatory authority. They complemented equine PK, GLP toxicity, and precedent preclinical studies, but overall they strengthen the evidence in human setup to support a clinical translation. And these data were part of the submission package that was accepted last year by the Swedish Medical Product Agency and the Swedish Ethical Review Authority, and contributed to the approved clinical trial of SYN321 that is now in phase 1/2A first in human study. And actually the clinical trial started this August. And here I just report the scan of the publication. These data are public, have been published. So if you are more interested in details, please go on to check the publication. And I'm, of course, happy to answer to any question. Thank you.
The ability to redesign the model for specific therapies is very powerful. From a drug development perspective, approximately how much time did it take for the redesign and revalidate the platform for injectable therapies?
Yeah, so in this specific case, it was quite fast because the redesign was mainly technological. So I would say that it took a couple of months for redesign and mainly for testing the biological model and confirming that it was working. But again, this was a pure technological modification. Of course, if the redesign also envision some biological modification or new biology protocol setup, that may take a bit longer. But in this case, was straightforward.
Cool. And then there's one really quick one I think you can answer pretty quick. Was the MPS data set alone submitted for the IND application independently or with in vivo animal data?
It was part of the of an overall application with also in vivo animal data. It was the only test to really prove the efficacy at the cellular level, but it was complementing equine and mouse data.
Great. Thank you, Paola.
Thank you.
So I'll go ahead and bring up Gareth from CN Bio. If you could go ahead and share your screen.
Yeah, thank you very much. So my name is Gareth Guenigault. I'm here on behalf of CN Bio, and I'm going to be presenting on basically how we used our organ-on-a-chip MASH model to test the efficacy of a small molecule HSD17B13 inhibitor, essentially how that led towards, a clinical trial and data being used as part of that clinical trial submission. So the key thing here to highlight initially is the kind of teamwork here. So this was very much between CN Bio ourselves and Innofarm. So who are we as CN Bio? We're an organ-on-a-chip company that we mainly focus on delivering human data using our BiomimX platform. We have a industry-leading liver model. Particularly, we have our MASH application that we use for exploring efficacy. And Innofarm are a clinical stage biopharmaceutical company, and they're focused on developing therapies for severe liver disease. And so they have this lead program, which is their HSD17B13 small molecule inhibitor. So a little bit of background on the disease. I won't spend too much time on this, but MASLD and MASH, essentially metabolic associated steatotic liver disease and steatohepatitis. Basically, these are fatty liver disease within the liver. So it's a buildup of fat that start to affect how the liver functions. So MASLD is incredibly common, it's about 30% of the world's population are estimated to have MASLD, is that buildup of fat in the liver, and in of itself it's not necessarily, you know, a negative thing. It does alter how the liver functions, and both MASH and MASLD are generally reversible through things like diet and exercise. But if that fat is prolonged and you get that fat buildup either through things like diet or genetic factors, then it starts to progress into MASH, and then you start to get really a lot more fibrosis and inflammation, and really in particular that fibrosis starts to damage the liver, and that's where you get the increased risk of things like cirrhosis, liver carcinomas, things that aren't reversible, and you know there really needs to be more drugs that target this specific disease. So why look at HSD17B13? So there's been a lot of data out there looking at cohorts of the population, looking at various different, genetic factors, and polymorphisms of HSD17B13 have been shown to be predictive. This kind of makes sense. It's very much involved in the lipid regulation. It's a lipid droplet enzyme, but essentially truncations in this or deletions even are related to having decreased risk of developing MASH and cirrhosis, decreased inflammation and fibrosis, and general changes in the lipids in the liver and the plasma. So in a sense, it's a really almost like obvious target to go and look at, because we know that if you knock this down, it has a protective effect against the disease. Now, the key thing here when InSphero was starting to look at this compound is that knockout mice don't generally produce that protective phenotype. So essentially they started with this for going, how do we explore whether this compound can have a anti-MASH effect? And so that's where they came and spoke to ourselves. This project started back in 2021 and we ran multiple projects for them. So a bit of background on our platform. So we have our BiomimX platform. Hopefully you can see my mouse, but essentially we've got our liver 12 plate, which we can run within the system. We can run up to 6 plates at a time with 12 wells on each plate. And the main aim of this is really to have the fluidic flow going through the tissue. So you can have your hepatocytes or other cell types from the liver seeded within this scaffold, which you see here, which would be in the bottom of each of these wells. And the media flow going past it introduces more nutrients, it introduces more oxygen to the cells as shear stress, and that means you get really functional hepatocytes. So as an example, you can see, albumin production, and then the data on the bottom right is data that the FDA published around our system, and you can see that, maintained metabolic activity in our system in comparison to things like spheroids and sandwich cultures. And so in particular, our MASH assay, how that works is, as I said, we take our liver 12 plate, we seed cells into there. So what we use is pre-validated primary cells. So that's primary human hepatocytes. And then we obviously add in Kupffer cells because they're essentially responsible for a lot of the inflammation you see as part of the disease. And then hepatic stellate cells, which are absolutely critical for the fibrotic side of things. We tend to use, when I say pre-validated cells, these are cells that we've used together in the past because it's really important to understand how those cells function. And we do tend to find a lot of primary human hepatocytes don't form good 3D tissue. We then introduce a dietary fat medium onto the cells, and we use that to drive the MASH phenotype. We leave them for a few days in that fat so they have developed that MASH phenotype, and then we start treatment with the therapeutic of interest. And then at the end of the experiment, we start to look at things like, steatosis, inflammation, fibrosis, and then general liver functionality, all the kind of hallmarks and things that we'd look for in a MASH disease. So in this specific set of experiments, we had a set timeline. So we generally pre-culture things like the stellate cells, set up the system, and then what we call day 0, we seed into the system our hepatocytes, the Kupffer cells and the stellate cells, which we tend to do in the 10:1:1 ratio. The day after that, we change to the hep-fat media, which is our, as I said, our proprietary media to induce the MASH phenotype. And then we leave it essentially till day 4, as I said, so that they can develop that phenotype before we start to treat. So on day 4, we start dosing with a test article and controls. So in this case, we were using elafibranor as a positive control. And then the drug we're testing here for InSphero, we were dosing every 2 days all the way up to day 20, where we ended the experiment. Now, it's worth noting the system is a closed loop system, so you have to do a media change every sort of 2 to 3 days. And so we're redosing the drug back into that system every couple of days. And everything here was done at triplicate as a minimum. Now, the experiment data I'm going to show, so there's essentially two phases to this. The first part was evaluating their initial compound they came to us, which was 678. And then the second part, which I'll only go into briefly, we started to screen compounds to actually, select the lead candidate to go forward from that point. So here you can see the general functionality and efficacy in a couple of different areas. So albumin for us has always been a good marker just to make sure that the cells are, there's no toxicity occurring, that the cells are nice and happy within the system and they're, performing like hepatocytes should. And you can see here that, in E678, both two different concentrations essentially match exactly the same as what we can see in our control. So in none of the work we've done here, in any of the compounds we tested, did we ever see any signs that there might be some negative effects on the cells. And then you can see markers such as oil red O, which was done at day 20 on the cells. You can see a reduction in that at the highest concentration of the drug. And similarly, things like IL-6 and inflammatory marker, you can see a decrease at the highest concentration of their compound. And then if you look at the fibrotic side of things, so we generally get to the end of these experiments and we fix the scaffolds and use confocal microscopy to be able to assess fibrotic markers. So you can see both collagen type 1 here and alpha smooth muscle actin. And essentially when we do this, we take our scaffold out and these are 3D tissues within that scaffold. And it can kind of look like they're slightly 2D within these pictures, but there's a depth to the tissue. And so we'll generally take, Z stacks through that scaffold and then look at the sort of intensity of the marker within that. And then we normalize it to Hurst, deliberately using that as almost like not quite a loading control, but to, normalize to the number of cells within each of those pores which the cells are sitting in. So we use that and then we can quantify the data and hopefully you can see it enough in the image here anyway, but you get this nice reduction in collagen type 1 and alpha smooth muscle actin with the 678 compound and you can see that significant in the data as well. So one of the really nice pieces about this work that I really enjoyed is, so we obviously did a lot of work on our side to look at some of the fibrotic and inflammatory markers, for example, but we also sent Inipharm a lot of the media that we took from the experiment. So every time we do a media change, we get about 1 milliliter of media, which gives you quite a lot to play with. And so we sent that to them and they were able to analyze it for, a range of different markers and across time course as well. So there's other bits I haven't shown here in terms of like, PCA analysis of markers over time and things, which is really interesting. But just to highlight a couple of key things here. So they measured things like the triglycerides, and you can see that they were significantly altered with their drugs. And these very much closely match what was seen in the plasma from patients. So these are two examples of a range of triglycerides. You can see the 44-1 here, which was decreased in plasma from patients with the protective allele, is also decreased within our model. And similarly, there were some that were actually increased, and we can observe that in our model as well. So this really closely, as I said, matches the data from people who have that protective allele compared to people who don't. And then going on from there, so that was kind of initial data that shared, yes, this compound does knock down MASH phenotype, it does reduce it. It has some beneficial effects. Can we then screen a range of compounds and see which one, has the greatest effect there? And so that led us around to a range of different groups of, similar versions of the same drug essentially, but, alterations to it. And what that led us to was 822. So you can see again here, just a slightly different way of looking at the whether it's causing any toxicity in LDH, but again, no sign at all when we went through this experiment of any indications of toxicity. And then again, if you're looking at the quantification of the confocal analysis of the fibrotic markers here, and there's a really nice knockdown in both collagen type 1 and alpha smooth muscle actin, better than we've seen in the previous version. And so that's what kind of essentially led us to go forward and Inipharm to go forward to essentially progress this towards phase 1 clinical trials. Obviously, now we provided essentially some of that initial proof of concept data almost to say this compound is going to work. And then that allowed them to go forward and do more work. So they also ended up getting in vivo data to be able to support this as well. So just to give a bit of a conclusion here, so we managed to generate that initial data that really demonstrated the efficacy of their compound, and that allowed us to go forward into subsequent studies that then helped select the final candidate for the phase 1 clinical trials. So we saw that decrease in fibrotic markers of MASH. We saw trends towards decreased steatosis and inflammation. altered triglyceride levels which match human data. As I said, there's other data that Inipharm generated as well around cholines and bile acid that also match clinical data. And then we were able to highlight, A22, which had similar results, but essentially greater efficacy than the 678. And as I said, it's now in phase 1 clinical trials, and I know we're all kind of looking forward to seeing what that data looks like. So just to say thank you, and particularly special thanks to Heather and Michael from Inipharm, who were an absolute pleasure to work with on this whole project.
All right, thank you, Gareth. So next I'll bring up Lizzy from AIM Biotech. You go ahead and share your screen.
Hi, everyone. It's a pleasure speaking with you today. I'm the Business Development Manager and the US Technical Lead for AIM Biotech, and I'm going to be sharing some of our data from our human 3D blood-brain barrier model that was recently included in a biopharma regulatory submission. So in case you're not familiar with who we are, AIM Biotech is an MPS company that was founded out of Roger Kam's lab at MIT, with our commercial operations beginning in 2015 out of Singapore, led by Kwan Chi Moon, who co-founded the company with the support of the MIT Singapore Alliance for Research and Technology, also known as the Smart Innovation Center. Over the past 10 years, through our own internal R&D, collaborations with Roger's lab at MIT, partnerships with biopharma companies, and in the hands of our customers, we have grown and expanded our product and application portfolios to include many different models of human health and disease, some of which are shown here. And we achieved this by enabling researchers to easily recreate robust human 3D biology, including things like perfusible blood vessels, obviously the blood-brain barrier, solid tumors that are co-cultured with immune cells, and vascularized 3D tissues such as organoids and spheroids. This in vivo-like biology is created using simple 3-channel microfluidic devices, which can be scaled from a 3-experiment chip all the way up to 40 experiments on an automation-compatible microplate. And our newest platform, which we call Organics, it enables larger tissue samples up to 2 millimeters in diameter to be injected through this open port to be then cultured in 3D tissue environments. And what's great about these platforms from a scalability and cost perspective is that there is no external equipment like plate rockers or external perfusion pumping systems that are required to achieve perfusion and oxygenation, because the bottom of all of our cultureware is sealed off with a gas permeable laminate. So although we have these different applications, today we're just going to focus on how our MPS was used to model the blood-brain barrier, or the BBB, to help assess off-target toxicity of a monoclonal antibody that was submitted in an IND file by a biopharma company. So as many of us are aware, the BBB is critical to maintaining brain homeostasis by controlling the transport of compounds in and out of the brain, and the specific interactions and the spatial organization of the 3 major cell types, the brain endothelial cells, the periocytes, and the astrocytes, which are what make up the BBB, are responsible for supporting that barrier function. Dysfunction of the BBB is implicated in aging and various diseases like Alzheimer's disease and cancer. And unfortunately, there's a very high attrition rate of CNS drugs, largely due to issues with crossing the BBB. And even if the compound being developed does not target the CNS directly, there can be potential off-target side effects. And so evaluating BBB transport is routinely conducted, typically using either in vivo models or more static in vitro models such as the transwell. There are some limitations to these models, however. So for in vivo, what can be challenging is that there are species differences and differences in expression of different transporter proteins. And there can also be challenges to actually isolating and investigating mechanisms of action. And then we know that the cost and time, and there are, of course, ethical considerations associated with running animal studies. On the other hand, the transwell BBB models often don't allow for the proper cellular interactions and spatial organization of the BBB to take place. So that can lead to non-physiological measurements of BBB transport, and then limitations on investigating BBB-specific active transport. And so, therefore, a biopharma sought to use our 3D human BBB model, since it does contain all 3 cell types spatially arranged with in vivo-like organization, and it is also perfusible and optically accessible. And relative to animal studies, our platform has a scalable throughput and is cost-effective, so making it usable for routine screening. To establish a human-relevant 3D BBB model, we rely on the self-assembly of the 3 BBB cell types in a 3D hydrogel to form a perfusible vascular network over the course of 7 days. And in this case, we are using the Identix platform, where a cell-laden hydrogel channel is in the center of the platform that is then flanked by 2 media channels that allow for perfusion and interstitial flow. The process of forming the BBB is called vasculogenesis, and it allows for the physiological formation of cell-to-cell and cell-to-ECM interactions that are critical for maintaining the BBB integrity. To validate the spatial organization of the cells, we use IF staining to measure expression of certain markers like CD31 to identify the endothelial cells forming these vessel lumens shown here in green, as well as phalloidin or F-actin to look at where the pericytes are. And so we see these shown in the yellowish orange that are clearly wrapping around and interacting properly with the blood vessels. And then we also use GFAP to stain for the astrocytes, which form nice end feet onto the blood vessels. And as you can see here, we also have some representative stainings for tight junction proteins such as claudin 5 and ZO1, as well as an efflux transporter protein, PGP. So all of this is to indicate that we have the proper spatial organization and morphology of our BBB networks in our model. Beyond morphology, though, we also wanted to confirm that the BBB barrier function is accurate and in vivo-like. And so we typically do that by perfusing fluorescently tagged molecules of different sizes through the BBB and using epifluorescent microscopy to capture images of the diffusion of the compounds into the brain tissue space over time, and then quantify the permeability of the BBB using these images. And in this particular publication, the researchers confirmed that the smaller compounds are able to pass through the BBB more readily than some of the larger compounds, such as a human IgG. And we see that when we quantify the permeability value of our model is on the order of what you would observe in vivo rodent models, whereas a transwell BBB model often has up to 2 orders of magnitude leakier barriers. We also confirm that our model enables the selective and targeted transport of antibodies that bind transferrin receptor, which is a common transporter used to facilitate drug transport across the BBB. And our results indicate that MEM189, which is a monoclonal antibody that specifically binds the transferrin receptor, can cross the BBB more readily compared to other similarly sized dextran, as well as a human IgG control, indicating a selective and functional barrier. Furthermore, the PGP assessment was conducted where we looked to inhibit PGP, which is an efflux transporter, as I mentioned, which when we do this, it can lead to an increase in the permeability of the chemotherapeutic paclitaxel. This is a substrate for PGP, and so when PGP is blocked, the efflux of paclitaxel is reduced, leading to the accumulation of paclitaxel in the brain tissue space and a correspondingly higher permeability measurement. This is certainly not all of our validation and characterization data of our BBB, but I do want to move on to the case study where this model was employed for the regulatory submission. But just quickly, before I do that, I do want to mention that we have recently developed a methodology with our co-founder, Roger Kam at MIT, to measure the permeability of unlabeled compounds, meaning that they're not fluorescently tagged. And so therefore, an image-based readout, like I just mentioned, would not be feasible. So to do this, what we instead do is we culture the same BBB cell types in the same manner in our organics platform, which has that open port on the top where a cell-laden hydrogel can be injected. A unique feature of this plate is that each of the 24 inserts can be removed, and the gas permeable laminate that closes off the bottom of each can be peeled off, which allows you to then extract the entire tissue intact. This could be then embedded for sectioning and histology, or if you're wanting to quantify permeability, you can actually digest that gel and run an ELISA or LCMS to quantify the concentration of the compound that has passed through the BBB into the tissue. And in some preliminary side-by-side comparison studies, we've shown that the results of permeability data that is retrieved from the fluorescence-based confocal imaging technique is very comparable, or is no different than when you use the gel digestion and the ELISA method. Another advantage of this is that when you digest the gel, you can also isolate the cells from the matrix and distinguish the concentration of a compound between those 2 samples. And so with all of that in mind, I now want to share how this 3D human BBB model was employed to support the regulatory submission. And so the background for this case study is that the biopharma observed a increase or an accumulation of a monoclonal antibody in the astrocytes of the rodent brain, despite the therapeutic target being outside of the brain. And so what they were wondering is, if there is binding to these astrocytes, do we see any sort of off-target neurotoxic effects, such as changes to the BBB integrity or permeability as a result of this binding? And so to support this work, what we did was we cultured our 3D human BBB model using our Identix 4D platform to first measure the binding of the monoclonal antibody to the BBB cell types, and then to evaluate the potential disruptions to BBB integrity and permeability. So first, to detect the binding to the different BBB cell types, we cultured these cells in both monoculture and triculture in the Identix 4D and used fluorescence microscopy to measure the co-localization of a GFP-tagged monoclonal antibody or a human IgG control, as shown here in these two images. The astrocytes in this case are tagged with an Alexaflor 647 GFAP compound. So then through quantification, using the co-localized staining, we observed that there was consistent binding of the monoclonal antibody to all three of the BBB cell types compared to the IgG control. We also saw that this was true in triculture, both after just 24 hours of co-culture, but also even after 8 days of growing the BBB. And we still see that binding to the astrocytes. And so what this means is that our model at least was able to recapitulate some of that monoclonal antibody binding to the astrocytes that was observed in vivo. To determine if that binding actually has an effect or it disrupts the BBB barrier function, we use the fluorescence-based method to measure permeability. And so to do this, we first treated the BBB with either the monoclonal antibody or the human IgG control. And then after 8 days, we injected a solution of 70 kilodalton fixed C dextran through the BBB and quantified changes in permeability. As a positive control, we also treated the BBB with a known inflammatory cytokine, TNF-alpha, at different concentrations and measured the changes in permeability. And what we saw through quantification is that with TNF-alpha, we see an expected dose-dependent increase in both permeability, but also with cell death as you increase the concentration of TNF-alpha. However, for the monoclonal antibody, as well as the IgG control, we did not see any increases in permeability or cell death, even when the compound, the monoclonal antibody, was administered at 100 times the therapeutic dose at 3,500 microgram per milliliter, which indicated that the monoclonal antibody, despite binding, did not actually disrupt the barrier function or the cellular viability. And then to further just confirm that there were no changes in BBB morphology as a result of the binding, we compared the vessel area of the BBB treated with the monoclonal antibody, IgG control, and TNF-alpha using immunofluorescence staining for the endothelial cell marker CD31. We saw no changes in the vessel area for BBBs treated with the monoclonal antibody. The only instance where we did see a decrease in vessel area was at the highest dose of TNF-alpha. And then similarly, for GFAP intensity, we did not see any changes when the monoclonal antibody was administered to the BBB. And so what this means in summary is that the data presented in the case study confirmed that despite binding to astrocytes in the BBB, the monoclonal antibody in development by the biopharma did not disrupt the integrity or the function of the BBB, which was critical evidence that was included in their regulatory submission. And so with that, I want to just thank you for your time, thank the researchers and team in Singapore for all their hard work on this project. Feel free to reach out to us or to myself, my email is here, with any other questions. I'll also be at SLAS in Boston in February, so if you're in town and attending, it would be great to connect. So thank you so much, and I'm happy to answer any questions.
Thanks, Lizzy. We do have one question for you already. Are these periocytes, astrocytes, and endothelial cells commercially available cell lines, or are they HIPSC derived? Are these BBB chips customizable with specific patient cell types?
Yeah, so the data shown here was with commercially available BBB cells. We have all of our protocols online with the specific cell types listed there. So these are primary, and happy to share those specific lot numbers and companies that we have purchased these from. We are working though on iPS-derived BBB cells because they're more consistent and reproducible. So those were not shown today, and those will be available soon. But yes, today these were just primary and commercially available. And then in terms of customizability, we're always happy to talk with you about what your needs are. If you have specific cell types in mind, we can work with you to optimize the conditions to grow a BBB that's specific to your needs.
Oh, thank you. There's also one, what are the various sources of the cells that are using? You might have already answered that, but.
Yeah, generally, things are going to be primary or IPS derived. So we usually are using primary, but those can have lot variability, especially the astrocytes. I feel like you're always ending up doing a bunch of optimization to try to get things to work each time you set up an experiment. And so that's where we are working on IPS derived cell sources that we can then provide to you. And actually, we are especially interested in providing kit-based types of solutions so that when you receive both the MPS platform, you also are going to receive the cell types that have been qualified and that are known to form the vasculature, but then also some of the ECM, because that can be a source of variability as well. So quality controlling all of that to provide you an assay-ready type of kit. Right now, we have that for angiogenesis and just general vasculogenesis, so like a general vasculature model. But our next up product in the cell systems line is a BBB kit.
Cool, thank you. All right, I think we'll go ahead and bring up the next presenter, James from Hesperos. Go ahead and share your screen.
Just really quickly, a lot of you are familiar with Hesperos, but just those of you who aren't, it was founded by Mike Schuler and I about 10 years ago, located here in Orlando, Florida. We've licensed a lot of patents from University of Central Florida and Cornell. We've got about 50 people here in our 14,000 square foot facility. We're a CRO. We do not sell our systems. And as I'll go through, we were the first microphysiological system to enable successful IND that enabled for rare disease with Sanofi, a phase 2 trial, which has now been successful and is now in a 2-arm phase 3. So what is it kind of we're doing differently than a lot of folks is I've been working this idea of clinically relevant functional readouts for about 30 years. And so what the idea is, you go into a doctor's office, he doesn't immediately stick a needle in your arm to be able to put up fluids, look at biomarkers, right? Looks how you're walking, how you're talking. Are you making sense? He listens to your heart. He checks your reflexes. He might ask about elimination. What he's really checking on is your muscles contracting okay. Is electrical activity of your neurons and cardiac cells functioning properly? Is your neuromuscular junction working well? Is the barrier integrity of things like GI tract and kidney working well? So what I've been doing is taking silicon devices, using surface chemistry to integrate cell systems with it. So I could say put cardiac cells on a thin piece of silicon. So when it contracts, it bends, it deflects the laser, I can get force. I can pattern cardiomyocytes to be able to get the conduction velocity, beat frequency, QT interval. Again, same thing with skeletal muscle. We have a wide variety of different neuronal types that I'll go through today, where we can look at function. We can still look at biomarkers, but we don't have to. This allows us to non-invasively look for acute, more importantly, chronic monitor these systems. We've published these 4-organ system out for 28 days with L'Oreal. And we're trying to do this all without cell death. We're not trying to use death markers as our primary endpoints. We really are trying to do function that is directly relatable to what you would see clinically. So what we can do, we can culture out the cells, each types of cells on their own chips. So we could have, say, liver here, cardiac here, muscle here, CNS here, cardiac, electrical here. Time it so that they can be assembled in the base of the platform. We can then put a gasket on top of it, so we have all the different cell types in the same recirculating media. Everything we do is in serum-free media. I banned the use of serum in my lab about 25 years ago. We can still look at all the advanced imaging technologies because we have a clear top. So say if you got your favorite cardiac organoid you'd want to see next to our cardiac systems on the chips, we can do that in our model system without too much of a problem. These are so far the drugs we repurposed into phase 2 clinical trials. CMT with Vanda, ALS with our Apellis, ALS with our Genix, myasthenia gravis with Dianthus, multifocal motor neuropathy with Dianthus, and CIDP with Dianthus, and as well as CIDP with Sanofi. And then these are the ones I can talk about, and I'll kind of highlight some of that data next. So what for the neuromuscular junction model, what we can do, we can take electrically and chemically isolated chambers for motor neurons and muscle, grow axons through the tunnels to be able to innervate the muscle. We had used this model system to publish first dose response curve for Botox back in 2018. We are now currently funded by the FDA and NIH to be able to create a drug development tool to replace the mouse lethality assay. This has been accepted for review by the iSTAND program right at the moment. We also can use this for disease models. We started looking at this in terms of ALS. We've differentiated out the motor neurons, muscle, and Schwann cells for multiple SOD1 mutations and TDP43, and then the motor neurons for C9 and FUS. In effect, what we can do, again, take the disease case motor neurons, say with wild type muscle. Again, you stimulate, you get a corresponding contraction. You see deficits with ALS, and we can reverse that. And again, sticking with this whole idea of clinically relevant functional readouts, I talked to somebody who's running clinical trials for ALS up in Boston, and I said, what are some of the tasks you have a patient do? And say, well, we have them do a task with their hands faster and faster. What happens? They start getting jitters, and they also lose grip strength. And we can reproduce that by stimulating the motor neurons faster and faster. And if we see skipping here, that's your jitters. We see loss of tetanus, that loss of grip strength. This is a published paper out of Advanced Therapeutics. Again, just showing that perfect fidelity and tetanus with the wild type, one SOD1 mutation, lots of skipping, partial loss of tetanus, another SOD1 mutation, less skipping, complete loss of tetanus, and with FUS, less skipping, complete loss of tetanus. And again, these are statistically significant. And again, in this case, we have a wild type muscle. You see perfect fidelity and tetanus, showing that the deficit is at the neuromuscular junction. Now, there's lots of data on these systems, but I'm kind of limited by time. I do want to highlight one work application we did with Apellis. Now, they have a drug, pegcetacoplan, which basically blocks the complement cascade. So they asked us, could we create our NMJ system and make it an inflammation model? So we had a microglia on the motor neuron side, and then Schwann cells, and either unactivated monocytes or activated monocytes on the muscle side. And see, you're looking at neuromuscular junction number. In the unactivated monocytes, we add in complement syrup, you see the deficit, and then an increase in MJ number with their compound. With the activated monocytes, you see a very large deficit, but then that's reversed. Now, Biogen had gotten a drug authorized for the SOD1 mutation just using neurofilament light data. So again, for this TDP43, they had done it for SOD1, we did it for TDP43. And again, you see the adding in the complement, neurofilament light goes up, and then it goes down in a dose-dependent manner. This was the only data utilized for Apellis for successful IND, successful ODD. Argenx utilized this model system in collaboration with some animal data to be able to get this drug into this clinical trial here. We also published the first dose response curve for an antibody, okay, with this system against the other antibodies not having any effect, being able to look at myasthenia gravis. So we looked at the anticholinergic antibody, and we wanted to, what we monitored was the different types of deficits that are caused by this, which is blocking, internalization, or complement deposition. Again, just showing two different methodologies in terms of looking at blocking versus internalization with either non-permeable permeabilizing cells or permeabilized cells. Again, without an antibody, we see about the same amount of receptor on the inside and outside. Low levels of antibody, we see it going down on the outside, and then high levels of antibody, we see this being downregulated. This is published in Frontiers in Cell Development Biology, and this was the only data utilized by Dianthus for this successful IND. And again, it's been successful, so they just raised $288 million to be able to do their phase 3. We were also approached by Vanda Pharmaceuticals for looking at a precision medicine application for a single patient. Charcot-Marie Tooth disease is a rare disease. CMT2S is a rare variant of that rare disease. So we took patient fibroblasts, turn them into stem cells. We differentiate them into motor neurons, basically showing that we get mature motor neurons in the system. Now, very interesting, CMT was misdiagnosed as ALS for years until they realized it doesn't have the jitters that ALS does. So what we did, we didn't see any changes in fidelity, but we did see changes in fatigue index at every time measured. And you see here, this is the fatigue index or the tetanus in the system. You see a different type of tetanus for the CMT mutation. And what you see here, what we saw was a chaotic tetanus, which is very interesting. It was a new phenotype we had not seen before. And they created an ASO, antisense oligonucleotide, for this specific patient's mutation. And you can see here it was reversed. And this was the only data utilized for the IND and the ODD for this particular patient who is now being dosed. And this was published in Molecular Therapy Nucleic Acids with Vanda Pharmaceuticals. This is now published at this point. We also create myelination models with this. This here is a publication where we induce pluripotent stem cell derived Schwann cells, myelinating induced pluripotent stem cell derived motor neurons. Again, all human. Again, we were able to use confocal microscopy to be able to create the G ratio to avoid doing TEM for that. Sanofi had seen these original models for myelination. We could create a conduction velocity model, which we did. We turned the NMJ system 90 degrees. We now run the axons over top of our electrodes inside the tunnels. We're able to use that to collect data. This is pre-compound testing with the untreated, the serum, patient serum plus an isotype control, patient serum plus their molecule. And again, you see after testing that the control, you still see a nice range of conduction velocities, or we see a far shift to the right with the isotype control, but their molecule basically was able to maintain that. This was the only data utilized for this IND that enabled this phase 2 trial, which is now in a 2-arm phase 3. And this is published in Advanced Therapeutics with Sanofi. Now, we've also been able to look at model systems for the CNS, okay, where we can create a model system for long-term potentiation, where we can pattern cortical neurons, where we can put one collection of neurons over top of one set of electrodes, another population here. We can simulate here and record here, reproducing dual patch clamp electrophysiology. This was published in Stem Cell Reports. And again, showing that we can actually have a correlation between the patch clamp for Alzheimer's with A-beta scrambled, A-beta oligomers, and A-beta plus donepezil, which is an approved therapeutic. And again, the key thing here is seeing a one-to-one correspondence with the high throughput MEA system between the A-beta scrambled, A-beta oligomers, and A-beta plus drug, which has now been published in Nature Scientific Reports for 4 different AD drugs. Now, what we can do, we can take this model systems here and create other multi-organ systems here in terms of Mike Schuller had created the first multi-organ system back in 2004. This is a more advanced system here looking at opioid overdose and recovery with off-target toxicity. But the main thing I wanted to show this for is that Mike had invented this idea of a pumpless platform. This is a cover of Lab on a Chip from 2010, where what we do, we use gravity to pump it. What that does is gets rid of all the external pumps, all the external tubing, gets rid of the bubbles. It also makes it a low volume system. So I can see the effect of both parent and metabolite in the same system. AstraZeneca had approached us. They had a compound that flew through preclinical. As soon as they put it into an animal, they saw a large QT effect. So we looked at the heart only, and we see a large QT effect for terfenadine, which is an antihistamine that was pulled off the market because it caused a large QT effect. However, it affects fenadine, marketed as Allegra, does not. And you see that shifted far to the right when we add in the liver. You see the conduction velocity was not affected at the liver present. IC50 at 33 micromolar, okay, in the heart-only system, and again, without affecting cardiac viability. So this was the validation study we'd used before we started looking at their proprietary compound. Now, what I wanted to show this for is because we can then take these model systems and create PK-PD relationships. We can account for absorption by measuring it in the system. We can account for metabolism. Again, since we actually have the clinically relevant functional readouts for the cardiac, in this case, QT interval, we can get the pharmacodynamics. Now, in this particular case, the molecule approaches the hERG channel from inside the cell. So we actually built in the transport into the cell to be able to get the actual pharmacodynamics. And then this is basically data we were able to extrapolate for guinea pig, dog, and non-human primate. Okay, that AstraZeneca had, and this was published in Nature Scientific Reports. We also worked another project with Vanda Pharmaceuticals. I don't have time to go into all the data from this, but we are working on a publication for it, where they had gotten a 3-month dog study for looking at toxicity. They were asked to do a 9-month beagle study for this. They did not want to do that, so we created a 4-organ system where we looked at their compounds toxicity with 2 different positive controls for each organ system. That data was utilized to submit to the IND, and that was accepted for review. And I think it's becoming close to becoming the finalizing of that IND. Again, this is for safety as opposed to the previous what I showed you for efficacy. We are now working with other companies to try and eliminate large animal model, at least one large animal model, if not all large animal models for safety evaluations, and those are being prepared for submissions currently. Now, so what I wanted to try to do is to look at our malaria on a chip model. Lastly,
James, you just have 1 minute left.
Okay, just very quickly, we were able to create a splenocyte in the endothelial cell liver cell system to be able to look at parasitemia, adding blood into it, and be able to look at the parasite life cycle. We were able to monitor these different parameters to get the active concentration. And so we were able to, for chloroquine, which is an anti-malarial, we were able to get the pharmacokinetic profiles, the pharmacodynamic profiles, to be able to predict maximum tolerated dose, no of 0 effect level dose, the EC50. And then it's very close to what they see in humans, and be able to have to predict the parasitemia for that. And this digital twin model was just published in Advanced Science. So hopefully I've given you a sort of a snapshot of a lot of different things we're doing. Happy to engage with you if you saw something of interest. You can come to our website or come into send information to here, and I'm happy to take questions.
Thanks so much, James. We don't have any questions or time for questions right now, so we'll go ahead and bring on the next presenter, Pauline from Alveolix. If you could go ahead and share your screen.
So thank you for the introduction and for the opportunity to present how Alveolix technology can be used for developing a new drug, and especially how the data we are generated can support regulatory application. So I will start by a general introduction about our technology, and then I will show you some concrete examples. So as you may know, Alveolix is a Swiss biotech company specialized in human model solution, especially organ-on-chip. We have developed a system called AXBarrier-on-Chip that allows us to mimic several organs, including their dynamic microenvironment. So typically we can reproduce the breathing motion of the lung, the peristaltic movement of the gut, the muscle contraction and relaxation of the bladder. And the system is actually composed of 4 key components. You have the IX-12, which is our consumable. It's where we are culturing our cells. The exchanger that allows us to exchange the medium inside the plate. The actuator, which make possible to recreate the mechanical stretch. And finally, the dock station that make the interface between the 2 pneumatic components and our consumable. if we have a look to the IX12 plate, it's been engineered to follow a 96 well plate format, making compatible with standard equipment. And it contains 12 independent models, and each of them integrate a ultra-thin, porous, and flexible membrane that is used as support for the cells. And the setup allows us to culture different types of cells and to adjust the complexity of the model in function of our needs. So typically, we can opt for mono, multiple culture. Here, for example, you have an air-blood barrier with immune cells. And to complete the microphysiological environment, the technology allows the introduction of mechanical cue, especially the 3D cyclic stretch. And for that, at the bottom of the plate, we have a membrane that closes a cavity. And this membrane can be pulled up and down. And this deflection will be transmitted to the IX membrane where the cells are. So you can see here on the video that you have the 3D deformation of primary alveolar epithelial cells. And in that way, we are reproducing the breathing motion. One thing which is interesting to know is we can tune several properties. So for example, we can adjust direction, amplitude, frequency, and so on. And overall, the chip is highly compatible with our current established readouts, so we can measure barrier function, notably via our T-ROX device. We can measure protein, cytokine release, gene expression. We can also do live fluorescent imaging. We can do most of the innovative omic readout like transcriptomic, proteomic, and the technology really allows us to help our customer to assess the safety, efficacy, toxicity of different molecules on a large panel of conditions. And today, I would like to share with you how it has been used to assess the safety of new immunotherapeutic compound. So basically, cancer immunotherapy cover all treatment that use immune system to fight cancer. So this range from monoclonal antibody to small cytokine that help to regulate immune response. And even if immunotherapy have a huge potential, it presents a high risk of immune-related adverse events, as well as a high risk of tumor toxicity. So this is why it's really important to assess the safety when we are developing a new compound. And the issue is traditional animal model have important limitation for that, mainly because the immune system are fundamentally different from ours. So to address this, one of our pharmaceutical partners aim to develop a human-specific translational safety strategy that rely exclusively on new approach methodology. So for that, they use about 21 human cellular systems to reproduce vital organs like liver, heart, kidney, and among them, our AXBarrier-on-Chip to detect off-target toxicity on the lung. And the data generated here were submitted to regulatory agencies, especially FDA and EMA, and the compound successfully progressed to the clinical phase. So yeah, let's see how we can bring data from bench to clinic. And the first step for that was to ensure that we was actually able to predict the safety of a compound in our model. And for that, we use FOLR1 TCB, a compound that shows severe lung toxicity on monkey. So the TCB T-cell bispecific antibody are antibody engineering to recognize both T-cells and the target, which is in this case FOLR1. And the antibody work by bringing close the T-cells to the cells that express the target molecule. This creates functional immune synapse that leads to the cell killing. And here, the 4-layer-1 is actually a molecule which is overexpressed in certain type of cancer like ovarian or breast cancer, but it's also expressed in healthy tissue like the lung and the kidney. So it really presents a high risk of on-target off-tumor toxicity. And we wanted to see if with our chip, we will be able to recapitulate this toxicity. So for that, we culture patient-derived alveolar epithelial cells on the membrane with immune cells. And we treated our cellular model with 4-layer-1 TCB and also with a non-functional TCB variant as negative control. So we firstly check the target detection in our model. And you can see here via immunofluorescence or gene expression that 4-layer-1 was well on the cells. And then we have performed the treatment on chip and measure several functional readouts, especially the alveolar barrier function. So you can see on the left that we observe a drop in barrier tightness of more than 70% with TCB at 0.1 microgram per milliliter. And on the right, we can see that at this concentration, we have the release of several inflammatory cytokines like interferon gamma, TNF-alpha, granzyme beta. And we also investigate the cytotoxicity, and we found that the TCB increased the apoptosis signal compared to the negative control. And also when we check for LDH release, we found that the higher dose of TCB caused membrane damage. The last thing that we have checked was the recruitment and activation of immune cells. And we could observe that in the presence of TCB, immune cells was specifically recruited to the epithelium. Also, when we have analyzed by flow cytometry the immune cells, we could find that both CD4 and CD8 were well activated in the presence of TCB. So overall, we saw that with this model, we can predict toxicity in the same way that what have been found in the monkey study. And then we can use our technology for evaluating the safety of a new compound, and notably MAGE-A4 TCB, which have been developed by our partner. So this compound targets MAGE-A4, which is an intercellular protein commonly found in many cancers, but which is normally absent for normal tissue. And the approach here was actually pretty similar to what have been done with 4-layer-1, except that as MAGE-A4 is inside the cells, the TCB recognizes the MAGE-A4 peptide when it's presented by the HLA-A2 on the cell surface. So here we have used our immunocompetent model with patient-derived alveolar epithelial cells, co-culture with immune cells. And as a positive control, we include ESC1 TCB, which is known for its cross-reactivity with off-target peptide. And as a result, we are assessed again a panel of readouts starting with barrier and inflammatory response. So here we saw that what with ESC1 or MAGE-A4 TCB, we didn't observe a significant difference in barrier disruption. However, when we had a look to the inflammatory response, we could observe a significant difference between the two compounds. So if you have a look to IL-10, granzyme B, interferon gamma, you can observe a significant upregulation with ESC1, our positive control, while the MAGE-A4 profile remain cleaner. Also, we have performed apoptosis and LDHSM. While we didn't see a significant difference in overall cytotoxicity between the different conditions, we observe a significant increase in apoptotic event with S1. In contrast, if we have a look to major 4 TCB, we have a safer profile independently of the concentration which have been used. And this is actually well visible in the fluorescent image that you have on the left, which highlight the dead cells. So overall, our results indicate that major 4 TCB didn't cause significant off-target toxicity in our lung model. And to give you an idea, the data were included in IND submission to several regulatory agencies, and the compound successfully advanced to a first in-human clinical study. In this trial, 22 patients with major 4 positive solid tumor received at least 1 dose, and it appeared that the starting dose was shown to be safe. So to conclude this talk, I have shown you how our technology can be used to assess the safety of a new immunotherapeutic compound. And by using specific human cells, our approach can overcome some of the key limitations of traditional animal model. And most importantly, our system can support more reliable drug development and regulatory decision making. So, okay, go away. So yeah, if you think that technology can be useful for you, don't hesitate to contact me. And with that, I would like to thank you for your attention.
Thanks so much, Pauline.
You go back.
All right. We can go ahead and transition to the roundtable now. So I'm going to bring all of the presenters up. Are the circuits in one chip considered technical replicates or biological? Might have also been Leopold. One of the first two.
I'm probably the wrong person to ask because we are one of the rare systems who don't use circuits. These are static culture, so I will leave
think
it
it was
to
for Leopold
others.
then.
Yeah, I mean, this question is always a mix. I think in a way they're in between. I would rather count them as technical replicates. I mean, usually, depending on the assay that is run, you can also include true biological replicates by including different donors, as long as it isn't run on a cell line, of course. But yeah, I think it's somewhere in between, but closer to the technical replicate, to my feeling and my understanding.
Great, thank you. And then this other one, I believe, was for Gareth. Is studying drug-drug interactions, CYP450 enzyme, a good application of the liver-on-chip models? And any of you have worked on or aware of workarounds for that?
Repeat the question, because that...
Is studying drug-drug interactions, parentheses, CYP450 enzyme, a good application of the liver-on-chip models? And have any of you worked or aware of workarounds for that?
I mean, yeah, part of the joys of having these, I think pretty much everybody's systems here, rather than having standard 2D models, is that you get better CYP functionality within, these systems. So looking at drug-drug interactions, you'll get more metabolites being formed and all that. They are good systems to be able to look into that. I can't say we've done it massively extensively. They tend to be quite complicated, but yeah. James, you're muted.
Thank you.
Yeah, I just want to add to that we've done, 20 different CYPs in our liver model. And we're working with a company called GalNAs-RX they have developed a platform to predict drug-drug interactions for CYP inhibition. That is actually a commercially available product that they use for pharmacists and clinicians that we're now bolstering that using our MPS data to make it more predictable.
Thank you. Bruno, did you have something?
Yes, so at least in liver microtissues, and I guess it's the same for most of the models here, the liver models. I mean, the CYPs are inducible and they are can be repressed. So on the basis of that, this could be a good foundation for drug-drug interactions. Yeah.
Thank you. Okay, this next question is for Pauline from Alveolix. You listed kidney as a model platform on your capabilities slide, but I'm not seeing this on your website. Can you clarify a bit further what component of the kidney this model reflects, glomerular or PTEC, or otherwise, when you will have data available to share publicly?
Yeah, so basically the kidney model is currently under development. So we have done already some study about it, but not on the one that I present to you today. Today was really focusing on the lung. The model will be soon on the website. We are currently making some updates. So yeah, everything will be available very soon on that.
Thank you, Pauline. Can the different platform manufacturers comment on challenges to quantifying drug exposure in their platforms and providing biological relevance to human exposure?
Well, I showed you the data we had that we published for the digital twin. We were able to predict maximum tolerated dose, no adverse effect level dose, and EC50 that correlated well with 3 different antimalarials. So we can use our in vitro platforms to do PK and PD to be able to predict in vivo outcomes.
Great, thank you. Bruno, go ahead.
Yes, so that's the complicated question. So I would say it's difficult because all our test systems are made with different components, different plastics, different material. And typically when we work at InSphero, we get, we tested about 1,200 different drugs last year. So we cannot know how each of these drugs are going to interact with the material. So based on that, I would say that the other very important aspect to it is you are going to have different measurements for the same drugs in different test systems. So if I can take a simple example, the simplest ever, cytotoxicity, you will have different IC50s in the different test system. That's because of how much media there is, how much circulation there is, how many cells you have, how active they are. So I would say how we approached it. So we published last year a paper where we tested 152 FDA-approved drugs in our test system. And then if you use always exactly the same test system and the same treatment condition, very standardized, then you can compare your drug response to other known drugs. And then you can draw similarities. Then you can, so we are continuously expanding this set of 152 drugs, but ultimately at the end, using this fixed test system, you can see drugs that have similar physicochemical parameter, and then this should help understand how the in vitro concentration translate to the in vivo exposure and what are the effects that you can expect from that. So I would say that the short answer is this kind of question, biological versus human exposure, this should be determined empirically using just a very large set of drugs. And then you can see which drugs have similar physicochemical parameter and then compare your own drug to this. That would be my easiest way to answer that.
And we approach it differently. What we do is we use HPLC-MS to actually measure the absorption in the systems. So then you can actually get the exposure profiles. So then
yes, so that's very true. You can do that, but not when you test 1,200 drugs per year. That's the problem. And a lot of our clients actually ask for this kind of throughput. So.
Thanks. Lizzy, go ahead.
You're still muted.
Oh, you're muted.
You're muted, Lizzy.
Thanks, everyone. Just to add to the conversation, I was going to say the way at least AIM is approaching kind of predictions for drug exposure in a human is by working in partnership with QSP modelers and PK modelers. So for example, a company Deep Kinetics here locally in Boston, but also talking with like MPS labs who develop these types of models really nicely. And so I think the more that we can take the readouts that we are getting from our MPS models and use computational models and in silico approaches to actually correlate that with other parameters that you can obtain from the clinic, and then cross-relate between these data sets what you might observe in a human. And not only that, but you need access to these types of compounds. So again, through partnerships and collaborations with biopharma to actually have these test suites of compounds. So that's where things like the 3Rs Collaborative, I think, is doing a nice job of making sure that we are facilitating conversations between all the stakeholders.
Great. Thanks, Lizzy. Lorna, go ahead.
I'm probably going to say much the same as other colleagues around the table here, but a couple of things spring to mind. First of all, I described a PDMS-based chip in the alveolus, which allows us to get stretch. The disadvantage, if you will, is small molecules, particularly lipophilic ones, will disappear off into the plastics. That's quite a concern for a number of pharma. But we have shown actually in a bone marrow model using small molecule drugs and again using computational modeling that we were able to achieve the clinical levels of exposure. We also showed it with other noxious substances as well. So it's not a given that just because you work on PDMS that you have those problems. But to supplement that and to address perhaps some of the other issues that are maybe that the person who asked the question is interested in, we also have now generated a sister chip to our chip S1. It's called R1. It's made of a different plastic, and like Jay was mentioning, we use LCMS to show that highly lipophilic molecules or molecules we've known in the past that have disappeared off into the PDMS, when run in the R1 system, we do not see that same phenomena occurring. So for me, it's about choice. Take Bruno's point, running LCMS is expensive, it's time consuming. You can't always do it for everything, but I think if the sponsor of the study, or indeed ourselves, if we were running a study, if that was critical to the data we were trying to generate, we would look to generate it with LCMS for the avoidance of doubt.
Yeah, cell-free controls are a great thing.
Thanks. Does anyone else want to comment on quantifying drug exposure before we move on? Okay, so the next question is to everyone. Was your MPS dataset submitted for the IND application on its own or together with in vivo animal data? According to the FDA's ISTAM program, organ chip-based DILI results that have completed all three phases of qualification can be used for IND submissions without the need for animal data. So if we just want to go through one by one to talk about whether they were submitted on their own or with data.
So I can start. I did answer that question at the end of my presentation, that in the example that I provided on the alveolus chip, it's a drug repurposing study. It was to open a clinical trial application. It was in unprecedented circumstances. And it was also known that there is unlikely or was unlikely to be an animal model of choice, given the fact that the animals are not usually susceptible to the SARS-CoV-2 virus. So in that situation, it was submitted without. Very clear position there. The questions seem to disappear off down the ISTAM route. I should remind everybody, we're the furthest forward in the DILI program. We're at the final stage of our DILI application through the ISTAM program. So if I'm not mistaken, I don't think anybody would be able to claim that they have got a DILI model through the ISTAM program and into use of INDs. But the ISTAM program is there, of course, to help make regulatory decision making more effective, and by qualifying through that program, that therefore becomes a tool that the regulators can use. So just to clarify that to whoever asked the question.
Thanks.
Yeah, and just to qualify that, I mean, there's a big difference between efficacy and safety. Okay, so all of our efficacy models, okay, the bar is lower for utilizing the system. So most of the work we've done for efficacy was only our data without animal data. Now, sometimes people had some animal data that was inconclusive. They came to us to be able to then generate that. Now, with safety, everything we've done with safety is in conjunction with an animal model, because they haven't quite, again, as Lorna has said, nobody has gotten these, through the ISTAM program at this point where, you're now qualified as a drug development tool. So for all the safety data we've done, it's in combination with animal data. Most of the efficacy data we've done is standalone by itself.
Yeah, thanks. Bruno, go ahead.
Yes, I mean, result can always be submitted to FDA independently on whether or not it is qualified as drug development tool. And in our case, so the story that I explained, it was submitted to answer the question together with the animal in vivo data. But the way it was done was to actually understand the response that the sponsor saw in dogs, the toxic response, and contextualize that for the human. So it avoided having to make more experiment in vivo in animals, and also following the in vitro testing, then hypotheses could be generated that could be tested back on the sample collected from the first dog study. So we are firmly in this refined and reduced part of the 3R in this case example. So it was complementing the in vivo data.
Thanks. Paola, go ahead.
Yeah. Sorry if there is background, I'm currently traveling, sorry for it. But yeah, as I mentioned already during my presentation as well, also our data were submitted in combination with animal testing, but as also Bruno underlined, also in our case, they were able to prove something that was not possible to prove in animal testing directly, because the pathology we are targeting, osteoarthritis, is really human specific and mechanical environment specific. So they were able to have additional data that they could not in any other case present to the authority. And this was possible also because it was an efficacy test. We were not entering in this case in safety testing.
Thanks, Paola. Lizzy, do you want to go ahead and answer? Oh, Gareth, either one.
Yeah, sure. I was going to say it's kind of similar to what a few people have said here, which is, data generated was obviously kind of fundamental in our study to be able to do that initial proof of concept, essentially to say this is going to work, and to be able to do that and then also help select lead candidate. But it was in conjunction with animal data to be able to demonstrate that as well. And I do think even moving forward, to be honest, the more we can have multiple models showing the same thing, that just gives you a better, basically a better package of data to be able to go into clinical trial submissions, whether that's animal and human models, or whether you end up with multiple MPS kind of systems together pointing in the same direction. At the end of the day, all of these systems, animals, MPS, they're all models, they're not humans. And the more data we have from various different systems, the better, in my opinion.
Yeah, thanks. Lizzy?
Yeah. So in our case, the observation came from an animal study that there was accumulation in the brain of their compound that wasn't targeting the brain, targeting a different part of the body. And so therefore they wanted to know with the human formulation of the monoclonal antibody, did it have sort of an impact on the BBB? And that's where the human model was helpful in identifying whether or not that was the case. So I don't know ultimately what was the final IND file package, but I would assume there was animal data included in that as well. And ours was just sort of answering that specific human question.
Thanks. Leopold, do you want to answer?
Yeah, so with others it was also similar to the others. So it was submitted as part of a full toxicology package, also including animal studies.
Great, thanks. Pauline?
Yeah, basically to the best of my knowledge, there was also some in vivo study which have been performed. On the study, they really aim to go with in vitro, ex vivo, in silico mainly, but some experiment was also conducted in vivo. After for the detail about the IND submission, not so aware about how much have been submitted on that direction, but yeah.
Go ahead. Thanks. Bruno, go ahead.
Yeah, just to elaborate a little bit on this matter, which is really important. So the thing is, the eye stent, if we imagine, okay, we have a test system going to the eye stent, you can submit without animal data eventually, but the eye stent is very specific for a test system with a certain context of use. So it's quite unlikely that at this point you would have a test system that would allow you to get rid of all animal testing. Maybe in the far future, when you have a lot of different drug development tool with context of use overlapping or contiguous context of use that would address all the safety questions, maybe that would work, but we are far from there at this point because there is a very specific context of use to each drug development tools in the ISTAN program.
Thanks. Okay, next question. For those of you using a hydrogel, are you using animal-based or animal-free gel? Does that have an effect on your functionality or accuracy of your model? Go ahead, Paola.
Yeah. So we do use hydrogel indeed in mostly all of our tests, and they are always animal-free. So either natural human-based hydrogel, or in some cases also some synthetic hydrogel to make also the process more standardized. Of course, we do it because we want to keep it relevant for the human model we are developing. But in any case, the testing and the standardization of the hydrogel is a big part of the work to make sure the entire setup is reliable and standardized.
Thanks. Anyone else want to talk about hydrogels?
I guess in our case, we use whatever is suitable to the model needs. Any type of hydrogel, whether it's natural or synthetic, is compatible. But typically for our experiments, we are using fibrinogen, which is cross-linked to fibrin with thrombin, as well as collagen. And so sometimes those are coming from animals. I forget what the last part of the question was, but does it affect something about it?
Does it have an effect on the functionality or accuracy of your model?
I would say, yeah, I mean, hydrogels you have to pay attention to depending upon what you are culturing. So if you are growing blood vessels, those can be tricky to grow depending upon what type of hydrogel you're using. And so there does need to be optimization at times. But between human and animal, I don't know that there's a big difference. It's just a matter of tuning it in terms of stiffness, oftentimes, and getting the right cell densities to form vasculature. In the case of culturing cancer cells, though, typically we're growing those in collagen type 1 hydrogels, which does come from the rat tail. And again, it's just a matter of getting the right stiffness usually to make the system function.
Thanks, Lizzy. Anyone else? Okay, we can move on. The next question. So comparison of drug concentrations to a known drug is something to be always included in the study design. In other words, how do we determine how many fold for higher and lower than Cmax has to be tested in vitro to get a good measure of biologically relevant effects? Bruno, go ahead.
Yeah, so based on safety, I mean, as I said before, principally for every test system, you have to determine your thresholds yourself. When you have a new drug candidate, you have no idea what is the relevant Cmax, what is mean. So the way we choose to do it is to just determine empirically which threshold of Cmax we have a reduction of viability or not. Is it a concern for the in vivo situation? So there is a lot of such studies that have been published in many different test systems. And they used more or less compounds, but that was always the same idea. Try to understand what threshold in terms of fold of Cmax they see a signal that's a concern or not. And then what they did is they ran some safe and toxic compound and they tried to determine the fold like this. What is first interesting is that to the best of my knowledge, the huge majority of the test system, you need to test supraphysiological concentration to have a finding that match the known in vivo effect for well-characterized drugs. So it goes from 20-fold of total plasma Cmax to 500-fold or 200-fold of total plasma Cmax. And then you can, using a specific signal, classify your drug correctly with a good sensitivity and specificity. So principally what you do is you look at your signal, you know which one are safe and toxic, and you try to see which threshold of the signal classify them correctly. So it's empirical. which I think it's a good way forward, providing that you have statistical power, which means that you must have many drugs that, and then afterwards you can refine your threshold. So for example, if you know that you have a lipophilic drug and there's this molecular weight, then you can select from this huge database of drugs that you know the response in this test system. I take only drugs that have a similar physicochemical parameters. So they're looking quite alike, and you see where the threshold is to classify properly the safe and the toxic. And then you determine the threshold empirically for your drug of interest. So this is how we approach it to address this very complex question, because the thing is, those in vitro test systems, we all claim they are physiological, but the truth is we are million light years away from physiological relevance. I mean, you just have to see it's a ball of cell in a plastic plate or tube, or you have a full human next to it. So you have to find this kind of a Rosetta Stone to translate what the model tells you to what it means for the in vivo. And at InSphero, we don't see any better way empirically to test many drugs. You know the safety in vivo, you see the signal in vitro, and you understand how that translates through numbers, testing a lot of compounds.
Thanks, Bruno. James, go ahead.
Yeah, we've been pretty successful in actually calculating in vivo, correlations with known and unknown drugs using a combination of in silico and in vitro work, because we can do the PK/PD in our model systems, and then since we have the pharmacodynamics in vitro, we can then predict the in vivo pharmacodynamics, and basically the IC50s. And if it's an unknown compound, we can model the pharmacokinetics based upon things like Simulations Plus or other something like that. Or if it's a known compound, generally there's human data that we can understand what the PK was there. So, and again, these are all published. Again, most recently the malaria-on-a-chip model. So we're finding a combination of the computational methods with the in vitro methods. And again, making it a low volume system is really important. Bruno was saying about having to go to super physiologic, because we get rid of all the tubing and the pumps and stuff like that, we can actually look at concentrations that are corresponding to, the concentrations you would be able to see in vivo.
Thanks. Does anyone else want to talk about this question?
Maybe just add in a different point there. I think, as we evolve the platforms and the technologies, we are going to learn how to make these systems more physiologically appropriate and drive better correlation to clinical outcome. And I think one of the simple things that we can consider, again, is if we're using protein in our medias, but we're working with small molecules, then we want to look at free drug exposure, because that's what's going to drive the effect, not the total amount of drug that was added. And when we get into different organs, so the liver, for example, is it appropriate to use a plasma value? Should we be looking at a hepatic value, whether that's a portal vein value or an other measure? So I do think, Jay said it just nicely there a minute ago. I think it's actually going to be a combination scenario, actually, that I think is quite exciting for this field, that we can combine what we already know and we've measured in systems, but we can start to apply that to some modeling and add some sophistication here so that we continually improve. So I think we've got to start somewhere, but I think it's very fair to say that there's room for continuous improvement across the board.
Well, it's actually funny, Lorna is exactly right, but what really drives a lot of the stuff that we do is cost. We always start off with a 7-point dose response curve, and then they look at the cost and go, oh my God. It's like, then you up doing a two dose, and then you expand upon it. So what we try to do is do the least amount of complexity at the least amount of cost initially. And then you add in that complexity as to answer the questions that they have. So ideally, we'd love to run basically at least a 5-point dose response curve, 2 logs up, 2 logs down. That's ideal, but that costs money, and it's difficult, in a system like InSphero, where you can do a lot of things, for fairly cheap. You could probably easier do that, but again, it has its corresponding drawbacks as well. So, but again, as something that Gareth said earlier, it's going to be a combination of our stuff, right? No MPS company is going to be able to do everything that people want. So it's going to be combinations of all our work, that answer the questions that they have, and which is great for the field, right? Because it's not like one company is going to be able to take over, the field. Everybody's going to have their own things they do well, things they don't do as well, and we're already starting to set up collaborative collaborations between the different companies. And I think you'll see more and more of that happening.
Thanks. Okay, we can move on to the next question. Have you used any of the models post-approval or during clinical trials to elucidate a mechanism of toxicity seen clinically?
I mean, I can probably nod at that, but not give a lot of information, because when we've done a lot of that work, it's client work, and trying to go into detail gets a little bit tricky. But we've certainly had situations where someone's come to us and said, we've got an issue here, we don't understand why, we weren't expecting it, can we understand the mechanistic tox of what's going on here? And I think that can often be a really strong point for most MPS systems, is actually we've got more of that complexity, and that allows you to really kind of evaluate why things might happen. And then even in that case, we might miss things, right? We might not have the right type of circulating immune cell present in the system, whatever. So a lot of this is really about understanding the model you're working with and what it can answer.
Thanks. Does anyone else want to comment? Bruno? Yeah.
Yes. When you go on the liver tox webpage of the NIH, you look at all these drugs that we take for years and years, and they are classified as hepatotoxic or safe. There is almost nothing that is known about the in vivo mechanism of toxicity. So everything that we know about the mechanism of toxicity of drugs is things that come out or from animal model in vivo testing, because you co-treat them and you make control experiments and you do this and that, or from in vitro test system. Whether it's the actual mechanism of toxicity in humans is unknown, because you cannot do testing in humans. So we should really keep this in mind. And this is a real headache, actually, for in vitro model, complex in vitro model developer like us. Because, for example, we have a client coming and say, we want to test mitochondrial toxicity. We say, okay, we can measure all these endpoints, and they say, prove me that it works and will be predictive to the in vivo situation. And so the best thing you can do is take drugs that you think cause mitochondrial toxicity, but actually you don't know it. Probably it's the worst example, this one, but because you actually don't know which one causes mitochondrial toxicity in vivo. So you don't know which are the positives and the negatives if you want to calculate the sensitivity and specificity of your test system and of this specific measurement. That's the very difficult issue. So the only thing you end up with is you cannot really test whether your test system can predict in vivo mitochondrial toxicity, because you don't know which one really causes mitochondrial toxicity in vivo. So you can just go down to what has been inferred by a set of animal experiments together with in vitro testing in the past, who build a coherent assemble of proof to demonstrate that it might be the case for the in vivo situation. So that's, and this, again, you just have to go to the liver tox webpage of the NIH. You see mechanism of toxicity of all these compounds. There are speculations, and at the end, there is always this one disclaimer line. The in vivo mechanism of toxicity is not known. There you go.
Thanks, Bruno. Paola, do you want to comment?
Yeah, I just want to follow up on what Bruno is saying, which I think is indeed a really huge problem for the field. And you talk about toxicity and toxicity compound. I think for efficacy, the situation is even worse because there, when we have to prove to a customer that the model is actually able to predict the effect of a drug to reverse or treat a pathology, most of the time there are no, let's say, compound that are not even available because nothing is able yet to reverse the pathology itself. So it's not only a lack of data, but also a lack of compounds themselves. So I think that the benchmarking compound and the availability of clinical data that can confirm the value of our models is one of the main bottlenecks of all the qualification path. And I see the problem for toxicity. We work with cardiotoxicity. We have the same problem. I see even a bigger problem for efficacy.
Thanks, Paola. Does anyone else want to comment before I move on to the next? Okay. Does anyone have experience in submissions of their NAMs to the European equivalent of a US IND submission, a clinical trial application? No.
Not yet.
Okay. All right, next one. Do you know if your platforms are used mostly for drug discovery or drug development? Not much NAM data are submitted to the FDA currently, and especially very little MPS data are submitted. Drug development data are more relevant to regulatory submissions. Bruno, go ahead.
Yes, so most of the drugs we are testing are at the discovery stage. So really large volume, simple endpoints, throughput. And then I would say that maybe 20% of the study we are performing are actually for drugs that are at the development stage, where we use much more complex endpoints, so transcriptomics, compare animal to human models, albumin, CYP induction, all this kind of question. So short answer is at both stage, but most of it is in discovery.
Thanks. Lorna, go ahead.
I'm really grateful for the person who asked that question, actually, and I'm not sure if on the line we've still got anybody from Charles River, but for those who were at the MPS World Summit in Brussels, they held a seminar early morning on the Wednesday morning, and they had actually conducted a large survey where they actually asked this question of a range of different pharma and biotech, and the overwhelming answer is that, the majority of the respondents are actually using the tools that were represented by those around the table for internal decision making, but very few of those translate into the regulatory submission. So that does tell you that they are interested in using that, and of course, and learning from it, and the question becomes what's stopping them from taking that final step, and perhaps with some of the regulatory policy shifts that have started to emerge this year, and we expect more in the first half of next year as well, that might change. But just for the avoidance of doubt, and again, I'm not sure if there's any regulators online, but one of the things is that they still would like to see that data if it's being used for internal decision making, although they have a term which is it's out with regulatory purview. Yeah. So I think that's perhaps disconnect, and I think it's a fascinating area, because if the measure of success of a technology has it been in an IND submission, we're actually maybe doing a lot of the platforms a disservice, because they have been incredibly valuable in the prior parts of a project. And having spent 20 years in pharma myself, I know how important it is to rank order candidates within a chemical series, which is part of your internal decision-making process, to end up with one candidate that you're going to put forward into a regulatory submission. So something maybe for us all to talk about in another forum, or perhaps if there's comments here, I'd love to hear them. But as I say, if someone from Charles River is on the line, it'd be great to hear a little bit more from them on that particular survey outcome.
Thanks.
But
Anyone
also
else?
along those lines of what Lorna was saying, you know, if you want to learn more about these systems and meet some of these folks in person and everything, please come to the MPS conference in DC the last week of May. Okay. You know, I expect to see probably everybody on this call there. If you're interested, if you don't know much about, you know, MPS, please come to the meeting in DC.
Yeah, and I'll second that as co-chair of the program committee. We're still looking for a few session chairs, so if anyone's interested, then just reach out to me offline, and we have some opportunities available for folks who want to do that.
I definitely echo, attending the MPS World Summit. That's a great place to have these types of conversations. In terms of drug discovery versus drug development and where maybe AIM is playing, it very much depends on what type of biology you're creating in our system. So our platform is amenable to really any phase of drug development, including drug discovery. And when you want to do higher throughput types of screening, that's where you're going to maybe use slightly less complex biological models. So maybe you aren't vascularizing your patient tumoroids. That maybe is where you're using immortalized cancer cell lines for something that's higher throughput and simpler and more cost effective. As you want to build a more complex model and add different features, add in immune cells or other stromal cell types, add in things like vascularization, that now you're going to be doing a lower throughput basis, and so that would be helpful for efficacy in the drug development phase. So yeah, we're in both areas, but it very much depends on what actually you are culturing.
Thanks. Anyone else want to answer this question?
I just add in that, these are more complex models, so they do tend to fall, InSphero is a bit of an exception here to a certain extent, but they tend to fall less into the screening phase and more into that kind of, complex side of things, trying to understand greater depth in biology, which tends to lend themselves either to kind of early discovery or that much more late stage development alongside animal models, that kind of thing.
Thanks. Anyone else? This is our last question, so I think we'll wrap up after this. Okay, we can go ahead and wrap up. Thank you all again for presenting and sticking around to answer questions. Thank you for everyone who asked questions. I hope this was really helpful, and I hope you have a great rest of your day. Thank you.
Thank you. Thank you.
Thanks, everybody.
Thanks, everyone. Great job.